Ok, buonasera a tutti, sono Daniela Marasco del Dipartimento di Farmacia dell'Università Federico II. A nome dei miei co-chair e di tutto il comitato organizzatore vi do il benvenuto al 50° congresso della divisione della chimica inorganica della Società Chimica Italiana, e soprattutto è un onore dare la parola al rettore, magnifico rettore Matteo Lorito della Federico II. Prego, rettore.
Allora, buongiorno a tutti, grazie per l'invito, è un piacere per me portare i saluti dell'Ateneo a questo congresso della chimica inorganica, della divisione di chimica inorganica della Società Chimica Italiana. È un piacere perché diciamo, questi congressi delle società scientifiche sono sempre dei momenti belli di confronto, ma anche un po' di riflessione su quello che le società scientifiche possono fare. Lo dico da past president della mia società scientifica, oltre che essere momenti in cui si presentano le attività svolte, ma soprattutto momenti in cui i giovani hanno la possibilità di presentare il proprio lavoro, sono anche momenti in cui come dicevo, si riflette su come vanno le cose nelle associazioni, diciamo, scientifiche. Ricordando che poi queste società scientifiche hanno anche a volte e spesso dovrebbero avere anche compiti di indirizzo politico, diciamo, sul tema che mette insieme, diciamo, i ricercatori. È un piacere avervi qui a Napoli e ringrazio i colleghi e le colleghe per aver organizzato appunto in Federico II. Siete in un posto particolare, lo dico per chi viene fuori Napoli, questo è il nostro centro congressi, ex facoltà di economia. Qui vedete, diciamo, intarsiate su questa scrivania di fronte a voi le arti e i mestieri che erano, diciamo, parte degli insegnamenti di economia, quando questa era appunto una parte, un'intera facoltà, un'importante facoltà, poi trasferita in un'altra sede. E sono contento che sia a Napoli, anche perché la Federico II sta vivendo, da un punto di vista puramente, diciamo, scientifico, perché oggi di scienza si parla e per fortuna si parla di scienza, sta vivendo un momento particolarmente importante, particolarmente significativo, che va dal momento celebrativo che è finito da poco, diciamo, che è quello dell'aver celebrato 8 secoli di storia. È inutile dire che la scienza chimica in generale ha contribuito molto alla storia scientifica della Federico II, quindi con figure importantissime, diciamo, la prima donna, diciamo, che ha avuto una carica importante, mi riferisco alla Bakunin, ma insomma figure importantissime che hanno fatto, diciamo, la storia di questo Ateneo. Ma poi è un momento particolare perché, insomma, con l'attribuzione dei fondi PNRR, Federico II ha avuto, diciamo, un momento di particolare floridezza, diciamo così, di particolare successo, perché è un po' il combinato disposto del 40% al sud e di grandi progetti di grande qualità da presentare, determinato che noi, insomma, abbiamo avuto in questi anni la possibilità di investire capitali importanti, diciamo, nelle scienze dure, diciamo così, ma anche in quelle morbide, diciamo, un po' tutte e due. Quindi questo è un momento molto importante, lo vediamo dal fatto che anche nelle classifiche internazionali siamo una delle poche, se non l'unica, tra le 10 università, quelle più quotate, quelle italiane, che sale la classifica, ma lo vediamo anche dal fatto che i ragazzi, i giovani, scelgono di venire in Federico II e noi li accogliamo. Infatti questo è un Ateneo, questo riguarda anche l'azione, diciamo, l'attività nelle scienze chimiche in generale. Oggi Federico II accoglie circa 600 RTDA, che è un numero molto alto, quindi noi siamo da questo punto di vista l'Ateneo forse più giovane d'Italia. Insomma, questo naturalmente rappresenta anche una grande sfida, quindi un momento di grande attenzione alla ricerca, di grande attenzione anche a finanziamenti per la ricerca, perché noi stessi finanziamo progetti interni, eccetera, ma anche un momento di grande sfida. Ed è inutile dire che temi come la chimica inorganica, discipline come la chimica inorganica, sono da sempre fondamentali, non solo per l'attività della ricerca, parlo di ricerca interdipartimentale, parlo di ricerca, diciamo, anche non limitata solamente all'area chimica, perché insomma, io sono un biotecnologo, ho fatto da sempre collaborazioni con i colleghi di chimica, perché in un modo o nell'altro questo come dire, si realizza comunque quando vuoi fare ricerca a un certo livello, ma anche e soprattutto anche dal punto di vista della formazione. Noi oggi offriamo circa 200 corsi di studio, se mettiamo insieme tutti i corsi di studio, cioè le lauree triennali, magistrali, quinquennali, le specialità mediche o non mediche, i corsi di dottorato, le academy e quant'altro, oggi siamo intorno ai 350 corsi come offerta formativa. È un'offerta formativa molto molto importante, della quale noi siamo assolutamente molto orgogliosi, anche perché, diciamo, fa il paio con la nostra politica di inclusione molto forte. Noi oggi, pensate, lo dico ai colleghi delle altre università, abbiamo fatto una scelta che consente circa il 55% dei nostri, il 60% dei nostri studenti, e stiamo parlando di circa 50.000 studenti, di essere in no tax area. Quindi noi abbiamo come dire, e dicevo, in questo nostro sforzo di offrire più corsi, quindi in qualche modo avere un'offerta formativa che consente ai ragazzi di individuare il proprio talento, di coltivare il proprio talento e di inclusività nell'accogliere più ragazzi possibili all'interno dell'università, ovviamente le scienze di base, le discipline di base, come la disciplina appunto della chimica inorganica, sono per noi fondamentali. Sono fondamentali per tante cose, anche dal punto di vista economico, perché non so quanti conoscono il tema del costo standard, è un tema molto importante per l'università e per il finanziamento universitario, ma sono fondamentali perché contribuiscono, oltre che alla conoscenza del settore, contribuiscono soprattutto a dare un metodo ai ragazzi su come affrontare discipline che diventano ogni anno sempre più complesse, sempre più ricche di conoscenze. E infine, ve lo dico con molto piacere, devo dire che noi qui abbiamo una compagine delle scienze chimiche di docenti giovani e diversamente giovani, diciamo così, molto, molto forte, molto, molto attenta, molto attiva. Sono quei docenti che uno come studente si vorrebbe trovare in aula, vale a dire quando tu entri in aula e trovi un docente che non è solo uno che ha studiato qualcosa su un po' di libri e te la racconta, ma è qualcuno che ha contribuito a generare quella conoscenza e quelle competenze che ti sta trasmettendo, e questo per noi è un motivo di grande orgoglio. Quindi io vi ringrazio, vi do, diciamo, un augurio di buon convegno, visto che avete un convegno anche abbastanza denso, guardate il programma, è un convegno denso. Se volete organizzare in modo che magari viene a piovere, così vi sentite un po' più tranquilli a star qui dentro, perché fuori è una giornata straordinaria, la vista è bellissima, però diciamo non ci distraiamo. E poi se tra una sessione e l'altra potete dare un'occhiata al nostro golfo, anche quello è parte, diciamo, del piacere di vivere. Grazie e auguri a tutti.
Ok, continuiamo, ringraziamo ancora il Rettore della sua presenza e continuiamo con il saluto da parte del Presidente della Divisione di Chimica Inorganica, il Professore Mario Chiesa. Prego Mario.
Ok, va bene, buongiorno a tutti e a tutte. Io ringrazio il magnifico Rettore per l'ospitalità in questa sala magnifica, una sede prestigiosa. che è quello che di meglio non potevamo sperare per celebrare questo evento, questo anniversario del 50° congresso della divisione. E vorrei altrettanto ringraziare, ma davvero con affetto, i colleghi Francesco, Anna Rita e Daniela e tutti i colleghi di Napoli che hanno contribuito a realizzare a organizzare questo congresso. È stato un lavoro enorme e ha dato i suoi frutti. Chimica inorganica è diciamo, affascinante per molti motivi, no? E forse il primo è che noi diciamo, non ci occupiamo solo di un elemento o di pochi nella tavola periodica, ma abbiamo licenza di studiare tutti gli elementi della tavola periodica. E questa conferenza ormai da 50 edizioni permette di trovarci e scambiare come dire, conoscenza e idee proprio sulle ricerche che portiamo avanti in questo settore, in tutti gli ambiti della disciplina, quindi dalla chimica di coordinazione organometallica, la chimica bioinorganica, la chimica dello stato solido dei materiali, con metodi teorici sperimentali negli ambiti dell'energia, della catalisi, della fotochimica e delle nanoscienze. E questo appunto lo facciamo da 50 edizioni, la prima risale al 1967. Era un mondo diverso e diciamo, la scienza è progredita e ci fornisce metodi avanzati per studiare gli elementi della tavola periodica, e lo vedremo in questi giorni. Ma io prima che noi ci immergiamo, no, nel programma scientifico, come ha detto il rettore, particolarmente ricco di questa conferenza, vorrei spendere un minuto o poco più per raccontarvi e dirvi due parole su il percorso che abbiamo fatto e che ci ha portato qui oggi a celebrare questo 50° anniversario. Ho citato il 1967, era in quell'anno il premio Nobel per la chimica fu assegnato a Huygens, Norris e Porter per i loro studi sulle reazioni chimiche su scale di tempi ultraveloci. Fu un anno importante per la scienza. Per esempio Barnard fece il primo trapianto di cuore, e per dirvi come era diverso il mondo, l'IBM creava il floppy disk, un oggetto che conteneva a stento 80 kilobyte di informazioni, quindi non avrei potuto caricare qui sopra la presentazione che state vedendo. Dal punto di vista politico il mondo poi era in subbuglio. Nel Medio Oriente la guerra dei 6 giorni portava Israele a occupare la Cisgiordania e Gaza, eventi di drammatica attualità. In Grecia ci fu il colpo di stato dei colonnelli, e su una nota più leggera in Italia Gimondi vinceva il 50° Giro d'Italia, quindi vedete che i numeri tornano, era un 50° anche quello. E proprio in quell'anno, 1967, su impulso del professor Croatto, ci fu il primo convegno nazionale di chimica inorganica. Io qui vorrei fare davvero un ringraziamento a un dottorando che sta lavorando nella sede della Società Chimica Italiana, facendo un dottorato in archivistica, e che mi ha aiutato a disseppellire letteralmente dalla Biblioteca Nazionale gli atti di questo primo convegno, Federico Berretta. Questo primo convegno appunto si tenne a Bressanone nel settembre del 67, fu organizzato dal professor Croatto. Lì vedete una foto dei partecipanti, furono una 60ina di persone, e se andiamo a guardare i nomi dei partecipanti ritroviamo molti dei padri fondatori della nostra disciplina. Troviamo Balzani, troviamo Corradini, troviamo Croatto, Malatesta, Renato Ugo e tanti altri. Da questa prima come dire, conferenza, primo convegno, diciamo, nacque, no? Quella che diventò poi l'Associazione Italiana di Chimica Inorganica, un'associazione che era al di fuori della società chimica all'epoca, e nella quale, però se guardate, se leggetelo statuto, al punto 1 dello statuto, si diceva che potrebbe in un futuro rientrare come divisione della società chimica italiana, cosa che in effetti avvenne. Avvenne nel 1975, questo è il resoconto della riunione dell'assemblea dei soci dell'11° congresso nazionale di chimica in cui viene istituita la nostra divisione. L'anno dopo, nel 1976, ci furono le elezioni e venne eletto il primo direttivo in cui il presidente era il professor Calderazzo. E da allora molti presidenti si sono succeduti, molti sono qui, alcuni sono qui in sala oggi e sono loro che hanno, come dire, fatto sì che questa comunità sia rimasta vitale, attiva e sia cresciuta, e diciamo a loro va naturalmente il nostro ringraziamento e a tutti noi direi buon congresso. Grazie.
Io voglio fare un brevissimo saluto. Come dicevo prima, io sono Daniela Marasco e sono uno dei 3 chair insieme al professore Francisco Ruffo, alla professoressa Narita Falanga, e vorrei rivolgervi appunto un caloroso benvenuto a voi tutti e di benvenuto qui a Napoli. Noi siamo consapevoli dell'importanza e dell'impegno che in qualche modo abbiamo preso in carico e ci auguriamo che il congresso possa adeguatamente riflettere la ricchezza delle tematiche di ricerca della nostra divisione e che possa soddisfare appieno le nostre aspettative. Perché la sede di Napoli? Il rapporto tra Napoli e la chimica è molto profondo, è radicato nella storia universitaria della città, come diceva anche il rettore, in particolare con la fondazione dell'Istituto Chimico già nel 1862. Che poi si è trasformato nell'attuale Dipartimento di Scienze Chimiche della Federico II. Durante quegli anni, come faceva riferimento il rettore, a Napoli hanno condotto la loro attività di ricerca alcuni scienziati abbastanza importanti come Maria Bakunin, pioniera della chimica organica e prima donna docente in Italia, e Francesco Giordani, suo allievo prediletto, che in qualche modo ridisegnò l'assetto universitario, la politica universitaria e soprattutto la ricerca scientifica sia di base che applicata e in collaborazione con la ricerca industriale. L'importanza di questi 2 nomi per lo sviluppo sociale e culturale di Napoli risiede anche nel fatto che il Comune di Napoli negli anni ha dedicato 2 vie della toponomastica della città. Venendo più vicino ai nostri giorni, a partire dal 1960, si respirò una vera e propria periodo di rinnovamento all'interno della facoltà di scienze nell'area chimica, soprattutto quando il professore Licuri fu appunto assunto e portò in cattedra qui a Napoli tanti studiosi importanti, tra cui ci piace ricordare il professore Liberti e il professore Paolo Corradini, al quale attualmente è intitolato il Dipartimento di Scienze Chimiche. E sappiamo tutti che Paolo Corradini in passato, prima di venire a Napoli, aveva fatto parte del gruppo di Natta, che nel 63 è stato l'unico premio Nobel per la chimica in Italia e che ha dato un grosso contributo soprattutto da un punto di vista strutturale. Quello che ci piace anche ricordare è che negli anni 70 l'Istituto Chimico è poi diventato il Dipartimento di Chimica, nella sede storica di Mezzocannone ha allargato moltissimo i suoi orizzonti culturali, soprattutto avendo grossa disponibilità di spazi, e questo ha consentito per esempio di aprirsi anche alle materie soprattutto biochimiche e in quest'ambito mi piace ricordare un nome che alla nostra comunità è particolarmente, e a molti di noi diciamo anche umanamente, è particolarmente importante, che è quello del professore Carlo Pedone, che è stato anche presidente della divisione e che non solo ha ricoperto un ruolo importante per la Federico II, ma anche dal punto di vista del Consiglio Nazionale delle Ricerche, in quanto ha fondato il Centro di Studi di Biocristallografie, di biocristallografia, scusate, e che poi di cui è stato anche direttore. Tra gli studiosi che hanno collaborato con il professore Pedone, sicuramente una menzione particolare va al professore Vincenzo Pavone, che ha portato e sviluppato tantissimo la chimica bioorganica in Italia, i professori Paolillo e Temussi, che per i loro studi soprattutto di caratterizzazione NMR. Tra gli allievi di Corradini ci piace ricordare i professori Achille Panunzi e Augusto De Renzi per i loro studi sulla organometallica del platino, e il professore Sigu, che ha lavorato nell'ambito della chimica di coordinazione con applicazioni sui cristalli liquidi, e ovviamente un po' più recenti il professore Michele Vacatello e il professore Vincenzo Busico, ai quali si devono importanti contributi nella chimica delle poliolefine. Per quanto riguarda la connessione tra la comunità chimica di Napoli e la società chimica italiana, mi piace ricordare il fatto che già nello statuto del 1914 la sezione di Napoli è una delle 3 soltanto sezioni fondatrici della società chimica italiana. E infine vorrei fare un cenno, un riferimento importante ai giovani chimici, che hanno come riferimento la nostra divisione e che con i loro progetti di ricerca innovativi dimostrano di aver raccolto le sfide della chimica per un futuro sostenibile in settori molto diversi e articolati, come emergerà soprattutto dalle comunicazioni orali delle sessioni parallele di questo congresso. È importante sottolineare il fatto che la SCI Campania, come altre sezioni in altre regioni, ogni anno organizza un congresso completamente dedicato ai giovani, sempre più partecipato e sempre più numeroso. Ovviamente ci auguriamo che possiate trascorrere una piacevole permanenza in generale a Napoli, come diceva il rettore, e che possiate ammirare insomma le bellezze della nostra città. A tale proposito volevo dire che oltre a essere il 50° anniversario, cioè, insomma il 50° congresso della nostra divisione, quest'anno ci saranno i festeggiamenti, ci sono anzi già in corso i festeggiamenti per i 2500 anni della fondazione di Napoli, che è ovviamente una città, come sapete benissimo tutti, millenaria, e che tra i simboli millenari ovviamente la nostra città c'è sicuramente Castel dell'Ovo, che ovviamente potete vedere, anche se un po' diciamo incerottato, perché appunto si stanno in rifacimento, la facciata in rifacimento proprio per i festeggiamenti che ci saranno a partire da dicembre, e che accompagna Napoli e i napoletani in tutte le loro diciamo situazioni più importanti, non da ultimo l'ultimo scudetto, e che ovviamente ci accompagnerà anche per questa sfida internazionale per l'America's Cup. Grazie a tutti e benvenuti a tutti. Allora io credo che possiamo aprire la sessione del congresso e io do la parola al professore Peruzzini.
Allora, buon pomeriggio a tutti. Spendo pochissime parole per ringraziare ovviamente gli organizzatori, doverosamente, non solo per aver organizzato il 50° congresso, ma anche per averlo organizzato in questa sede, che è veramente una sede particolarmente, direi, prestigiosa, particolarmente adatta, e purtroppo le finestre sono chiuse, ma insomma la posizione l'avete già vista, siamo veramente nel cuore di Napoli, nel centro di una delle più belle città del Mediterraneo e quindi del mondo. Quindi grazie a Francesco, grazie alle 2 collaboratrici, Marasco e, mi sfugge il nome? Grazie. Bene, a me spetta l'onore e l'onere di introdurre la medaglia Sacconi, se posso avere le slide, perfetto. La medaglia Sacconi è ormai una tradizione della nostra divisione di chimica inorganica, a cui recentemente si sono affiancate, diciamo, altre prestigiose medaglie. Questa probabilmente è la più vecchia ed è la medaglia, diciamo, che la scuola fiorentina di chimica inorganica ha voluto, in collaborazione con la divisione di chimica inorganica, istituire per ricordare il maestro Luigi Sacconi. Luigi Sacconi fu uno dei grandi della chimica del secolo scorso, operò nella seconda metà del Novecento, e i nomi di Malatesta, di Croatto e di tanti altri chimici inorganici, Cambi ed altri ancora, Brunori e così via, riecheggiavano nei suoi laboratori alla fine degli anni 90. E qui vedete due fotografie di Sacconi, una è con diciamo con Fred Basolo. Fred Basolo è questo, forse anche più giovani sanno chi era Fred Basolo, uno dei grandi numi della chimica inorganica, dello studio dei meccanismi di reazione in particolare, un americano ma di origine fortemente italiana, piemontese come diceva lui. Io ho avuto il piacere di conoscerlo nei suoi ultimi viaggi a Firenze fino alla fine degli anni 90, e qui lo vedete insieme a un altro nome tutelare della chimica inorganica italiana, che è il professor Lamberto Malatesta, a cui doverosamente la divisione ha istituito una nuova medaglia a partire da 3 o 4 anni fa, penso. E nel 1996 venne istituita la Fondazione Sacconi. Le notizie sono ovviamente presenti sul sito web della Fondazione, chi ha voglia e tempo lo può vedere direttamente. Lo scopo è quello di ricordare la figura di questo grande scienziato e di tributare tutti gli anni praticamente un riconoscimento a un chimico italiano o straniero che si sia particolarmente distinto nel campo della chimica di coordinazione, che era della chimica inorganica in senso lato, della chimica dei metalli, che era il terreno, diciamo, di lavoro del professor Sacconi. Questo è il board attuale della fondazione e questi sono praticamente i premiati, e la storia di un premio la fanno i premiati, e vedete che la lista dei premiati è, a mio parere, impressionante. Alcuni hanno avuto il premio Nobel, altri spero lo avranno, e non sto a commentarli, ovviamente, li vedete da voi, sono nomi di enorme importanza. Scusate, sono andato fuori simmetria. Comunque la lista è impressionante e oggi mi fa particolarmente piacere annunciare quindi il premiato di questa edizione, che è la professoressa Geneviève Blondin del Laboratoire de Chimie et Biologie des Métaux del Comitato per l'Energia Atomica francese, con sede a Grenoble, per i suoi significativi contributi alla chimica di clusters biologicamente rilevanti, quali sono i cluster ferro-zolfo, che diciamo, ci hanno permesso di ottenere importanti informazioni non solo nel settore della bioinorganica ma anche della catalisi. Insieme al presidente Mario Chiesa vorrei quindi chiamare la professoressa Blondin a ricevere la medaglia Sacconi. Mario. Congratulazioni. Congratulations. I've spoken in Italian, but I suppose that you understand a little bit of Italian. Tiny, okay. So,
but I understand my name at least.
Okay, that's the medal. If you want to show to the people, just to take a picture, and let's say just a fully deserved medal, and you will continue the tradition of a great scientist in this. So, formally, thank you so much. Let's say.
Thank you. Very much.
And now I formally introduce you as a speaker, and I move to English. I have received a very short CV. I will just use a few minutes more. Professor Geneviève Blondin is obtained his PhD at the Laboratory of Inorganic Chemistry in Orsay, in France. That laboratory was directed at that time, let's say, by a very famous French scientist that was Professor Olivier Kahn. Professor Olivier Kahn was a good friend of many Italian scientist, particularly working in the area of magnetism. And many times he came to Florence, hosted by Dante Gatteschi, Ivano Bertini, and the other people of the school of Professor Sacconi at the time, and I remember him quite well. Later on, he defended his PhD thesis in 1992, that's correct, and then he became researcher at the Centre National de la Recherche Scientifique, sorry for my French, and he was moved back to Grenoble in January 2006 and became in the laboratoire of physical chemistry of metals in biology, that is currently directed by Professor Jean-Marc Latour. And she got important recognitions and put together a very active group of biologists, protein chemists, synthetic chemists, and spectroscopists, developing their research to provide new insight into the unique question of the physiological function, structure, chemical reactivity, and regulation of complex biological systems transporting and utilizing metal ions, and the exploitation of this knowledge, to develop bio-inspired systems with similar structures and functions. Let's say, she will have many interactions with important teams, including that led by Carl Wiegert, and after the beginning of his career, specialized in electron paramagnetic resonance spectroscopy, she moved to consider Mossbauer spectroscopy, especially of iron, and I think that today she will talk a lot about Mossbauer spectroscopy and of iron, indeed, especially that associated with the iron sulfur clusters. And the title of the lecture is just that one, Mossbauer spectroscopy, okay, you see there, and iron sulfur clusters, a still ongoing story. And let's see together where this story is going on. Thank you, Geneviève. Welcome to Naples again, and the stage is yours for the lecture.
Well, thanks a lot for this distinction. I think indeed it's a strong support for women in science. I never expect to receive a medal one day, I must confess. And well, as you've heard, I'm more a spectroscopist. I've spent some time on EPR spectroscopy when I was near Paris, but now, since I am in Grenoble for 20 years, I've moved towards Mossbauer spectroscopy, and I would like to tell you that indeed it's a spectroscopy that is very informative. So it has been discovered, this technique, since about 70 years ago, and there's only about 80 nuclides in the periodic table that present the Mossbauer effect. But one of the elements you can study is iron. It's very interesting because it is one of the most abundant elements in the Earth's crust. So it involves many scientific domains, and I'm more interested in chemistry and biology, as you may guess. And however, this technique is specific of the 57 isotope of iron, which is only abundant at 2%. So it may be a problem for proteins. Because I anticipate you're not all familiar with this technique, I've briefly summarized how it proceeds. You observe, in fact, a nuclear absorption, a nuclear transition, and in the case of iron, it's between the ground state with a nuclear spin 1/2 to an excited state with a spin 3/2. And to make this transition, you need to have gamma rays. They are emitted by the same type of nuclides that is formed in a nuclear excited state in the radioactive source, because you've got the disintegration of the 57 isotope of cobalt. But reality is always more complicated. And in fact, there is a quadruple interaction in the excited nuclear state, which split the level at least in 2 levels. And because you've got a fixed radiation energy, you have to modulate it. And this is done by Doppler effect. And you apply vibrational movement to the radioactive source. And that's why you've got spectra that are reference versus velocity expressed in millimeter per second. A few millimeter per second up to 12 generally allows to see all the nuclear transitions. And there are two main parameters you may extract from that. It's the separation between the two lines. It is called the quadruple splitting, and also the central position, which is called the isomer shift. And this parameter allows you to have many informations on the iron. The oxidation state, the spin state, you may have also information on the ligands, their coordination number, and because the nuclear spins are not zero on the iron nuclei, if you've got unpaired electrons, you generated a magnetic field that experiences the nuclei, and from the hyperfine interaction, you also may extract information about the spin coupling topology. This technique is specific of the 57 isotope of iron, but you will see all this kind of nuclei you've got on your sample. And so it's very complementary to EPR spectroscopy. EPR spectroscopy will be restrictive to half-integer spin system, but Mossbauer spectroscopy, you will also see diamagnetic systems like I will show you in the next slides. And because each type of nuclei have a different response, you may identify families, and more importantly, you may quantify the iron distribution. So I hope to, through the presentation, to illustrate a few points of this technique, and I will apply it to iron sulfur clusters. The most common iron sulfur clusters you may find in proteins or in enzymes are dinuclear, cubane-3 or cubane-4 cluster. The iron are bridged by sulfide. They are tetra-coordinated generally, and the coordination sphere is generally completed by the tolite function of a cysteine. They are involved in electron transfer reaction, catalysis. They can be also structural. Well, they play many roles. There were a lot of studies from Mossbauer spectroscopy, and I've listed some people who have done a very nice job. The main contribution is to, in fact, identify the different signature coming from the different clusters. Mossbauer spectroscopy allowed to evidence the delocalized valences within these structures. They are made mainly of iron 2 and iron 3, but you should talk about iron 2.5 plus. And from the magnetic properties, it was a delicate balance between, in fact, the electronic delocalization and the exchange interactions. Iron sulfur clusters have been studied in proteins originating mainly from prokaryotes and eukaryotes, but they have recently been identified in viruses. They may play a structural role, like in the first iron sulfur cluster discovered in bacteriophage lambda, but they also may be critical for the replication and transcription of the virus that has been evidenced in the coronavirus SARS-CoV-2 that we have all heard about, I guess. In 2003, there is a new class of viruses that have been identified called giant viruses. In fact, it was thought that viruses were small particles, and you use 2 nanometers diameter pore size filters to isolate them. But if you have larger particles, they remain in the supernatant. And that's what happened to giant viruses that had been discovered. You may see the large particles here that have been identified. They contain double-stranded DNA. They have many base pairs. They codes for hundreds to thousands of proteins, but the main puzzling discovery was that these proteins are unknown. They don't share any homology with known proteins from the cellular or viral world. The first giant viruses that has been identified was mimivirus. For microbe mimicking virus, it is this large 500 nanometer particles. And Chantal Arbergel in Marseille, which was involved in this discovery, investigated the infection cycle. And she discovered that in the late phase, there was a gene which is in fact the most transcribed gene that encodes for a very small protein, but very rich in glycine and cysteine, which is absolutely unusual. So that's why we have no predicted structure nor function. And she managed to purify this protein by fusing with the tag is-cis sumo. And when Benedict Berla, which is specialized in iron sulfur biochemistry, saw the color of the sample of the isolated proteins, she said, oh, you must have iron sulfur cluster in there. I will demonstrate it. So she started investigating, and the first thing she had done was to record UV visible absorption. And you see indeed features at 400 nanometers that are very characteristic of dinuclear diferric cluster in blue. But there are also features at higher wavelengths that are more characteristic for free nuclear 4-iron sulfur clusters that had been synthesized. And she also recorded EPR spectroscopy at 9 gigahertz and low temperature. And the only signal she detected was at low field. And this type of signal is really reminiscent of advantageous iron-free that you don't want to see. But it is also consistent with the trinuclear linear structure because of the antiferromagnetic interactions between the high-spin ferric ion. You get a spin 5 half-ground state. And for the same reason, but on the dinuclear species, it is diamagnetic, so EPR silent. So she called me to confirm this composition of the sample. So I turned to Mossbauer spectroscopy, and the first spectra that are used to record is a low field and low temperature spectrum. And you see a very nice doublet, and indeed, the lines you observed at 0 and 0.5 millimeter per second is very characteristic of the oxidized diferric disulfide cluster. But no sign of the linear complex. Well, yes, there is. If you look carefully, there is a weak absorption between -6 and +6 millimeter per second. And if you record the spectrum at a higher field, here 4 Tesla, you have the proof of indeed the composition of the sample. In the central part, you've got the signature of the diamagnetic diferric clusters with an isomer shift value that is fully consistent with ferric ion in a tetra coordinated environment with sulfide. And the two outmost lines that you observe at close to -5 and +5 millimeter per second, indeed is characteristic of this linear topology. You've got similar isomer shift compared to the previous case. And more importantly, you see you have now an upper-fine interaction, and you've got negative, positive, negative sign, which reflects the antiferromagnetic interaction between adjacent iron. So indeed, by Mossbauer spectroscopy, we were able to prove that the sample composition was made of diferric and triferric clusters in a ratio roughly 1 linear for 4 dinuclear species. The linear structure is very unusual as an active site of proteins, and the discovered virus was in 2003, but since then, Chantal Abergel and Jean-Michel Claverie discovered many other giant viruses, from Mimi, Pando, Pito, Molly, Tupon, and so on. They are huge, as you can see from this side. They are found everywhere in all waters around the Earth, in the Siberian permafrost. And there are many questions on these giant viruses because they are smaller analog. They don't have any iron sulfur biogenesis machinery as we have. So the question is how they get on the iron sulfur cluster from their host. In the case of the small proteins, I've shown you what is the function. It seems not to be critical for the replication of the virus, but it should have a function, and we are still working on that system. The other study I want to show you is when you investigate whole cell. Well, In the example I will tell you, the protein was not isolated in a too high yield to allow Mosbora investigation. In fact, we are quite demanding and asking for high concentrated sample. You may have also doubt on the reconstitution procedure because you may isolate the protein, but during the procedure that lost the metallic site, so you add again the metal ion to reconstitute, but you may not obtain a physiological active site. There may be a debate on the nature of the active site of the protein, and that was a very nice collaboration I had a few years ago with Lucia Benchi on the anamorphine. Or as Paul Lindahl did in the States, you may want to investigate all the iron homeostasis within the cell, and he spent at least 2 decades on investigating Saccharomyces cerevisiae. For the general procedure, when you want to investigate a specific protein in whole cell, you overproduce it in Escherichia coli with introducing a 57 iron salt in the culture medium, as you will do if you want to isolate the protein. But you will record the spectra on this overproducing cell and compare it to control cells where the protein is not overproduced, and you expect to detect an additional signal that you will attribute to this overproduced protein. And the study I will tell you today is on an unsubsiding protein that is found in DVH, and it's again a collaboration with Bénédicte Borla and Corinne Aubert again in Marseille. DVH is a sulfato-reducing bacteria. It is present in anoxic habitat, like waters and also our bowel. So at the end, the sulfate is reduced into hydrogen sulfide, which has many drawbacks. It is responsible for the toxicity of water. It may be also involved in corrosion of pipelines, and it may be also involved in Parkinson's disease. The analysis of the genome of this bacteria allowed the identification of proteins that were important for the anaerobic life. And one of them is ORPR, which is a regulator. It allows the transcription, the hydrolysis, of ATP into ADP. It is in a very, very reducing medium. And when you have this hydrolysis, the transcription is initiated. But if you raise the redox potential of the medium, it's not a reaction with dioxygen. It's really sensitive to the reduction potential. There is no more dihydrolysis of ATP, and the transcription is off. This regulator has 3 domains, and one is a sensory domain belonging to the PAS family. And there was the Bénédicte and Corinne had a strong evidence that there was an iron sulfur cluster in this PAS domain, and they asked me to confirm it by Mosbora spectroscopy. So we turned first to overexpression in Escherichia coli, but if there is an iron sulfur cluster, you better reduce the interaction with the iron sulfur cluster machinery of Escherichia coli. That's why we deleted the Ischia strain. We work in strict anaerobia because we are investigating a protein originating from a sulfato-reducing bacteria. We have the protein in the plasmid, within the plasmid PGF119, and not if the plasmid is empty. The EPR spectroscopy only allows to evidence by difference a radical. So if there is an iron sulfur, indeed it is EPR silent. If you turn to Mosbora spectroscopy, as previously the low field, low temperature spectrum, in black you have the control cell, and you see 2 intense lines. And in fact, it is well known in Escherichia coli, it's the doublet originating from the iron 2 pool, because you are performing culture in a strict anaerobia, so you don't have any oxidation. But if you look now to the overexpressed cell, you see that you have an increase in intensity at 0 and 1, and this is indeed characteristic of 4 iron 4 sulfur cubane cluster in the 2+ state, where the isomer shift is now higher, 0.48, because you have a mixture of iron 3 and iron 2. But even if you have 4 iron, you see you've got 1 doublet, that is 4 equivalent iron. So in Escherichia coli, fine, we've got a 4 iron 4 sulfur cluster. But what about DVH? In the bacteria, the active form is generated when you are in strict anaerobia, and this is a regulator. So you've got, it controls the simultaneous expression of two divergent operants, 3 to 5 on the left and 7 to 9 on the right, that encodes for protein, ORP4 on the left and ORP9 on the right, but I will only show you the Western blot analysis with the protein 9. But we have empty cells while you delete, you inactivate the ORP gene. You have not the regulator, nor the protein ORP9, but if you activate, you overproduce the regulator, you have both. The APR spectroscopy only allowed to evidence radicals, low-spin ferric system, and the reduced 4 iron 4 sulfur cluster of APase reductase, which is well known in this bacteria, but no evidence from, if you look to the difference between the control cell and the overproducing cell, of a signal that may be attributed to an iron sulfur cluster. So once again, it may be EPR silent. If you turn to Mosbora spectroscopy, you will then see that you have an important increase at 0 and 1, which is the position you expect when you are dealing with a 4 iron 4 sulfur cluster. However, in that case, to perform the difference is a little bit more tricky, because you may see that you have an extra absorption on both sides of this doublet. And in fact, this is due to the fact that you are releasing hydrogen sulfide, because you're dealing with a sulfato-reducing bacteria. And the hydrogen sulfide will react with the ferric salt you have introduced in your medium. And you get a mixture of iron sulfides. And as you see all the iron you have put in your sample. The first samples, indeed, we have investigated. It's shown here on light green. You saw a huge contamination by these iron sulfides on the overproducing cell, but after huge work, Benedict and Corinne managed to remove most of it. And if you compare these two spectra, you manage to see that by difference, you've got indeed the characteristic signature of a 4 iron 4 sulfur 2+ doublet. You may compare this doublet with the one I've shown you in Escherichia coli, which is there is no hydrogen sulfide that has been released. The doublet is slightly different, and the main difference is the line width. It's larger in DVH, and this suggests that there are a larger distribution of iron sulfur cluster into this overproducing cell. And this may be due to the fact that when you overproduce the regulator, you also produce protein, ORP4, 9, and 3. And unfortunately for me, they also contains 4 iron 4 sulfur clusters. Well, but my Mosbora spectroscopy we managed to evidence that there is indeed a 4 iron 4 sulfur cluster in this regulator. We don't have any X-ray structures, and AlphaFold predicts a free coordinated cysteine, but we don't know what is the force ligand, so that's a question we want to answer. And my colleagues suspect that indeed, because the iron sulfur cluster is in the sensory domain, it may change upon changing the redox potential of the cells. So there may be a change in the coordination, and we are currently investigating also the inactive form of the regulator. And in the last part of my talk, I would like to turn to synthetic iron sulfur cluster, because there have been a lot that has been synthesized before, and especially by the group of Richard Holm in the States. And I just mentioned a few results. And in fact, for example, the linear 3 iron 4 sulfur complex, it was my PhD director work when he was a PhD student, postdoc student. But however, a few years ago, there was some remaining challenges that remains. For the cubanes, for example, they were synthesized in all different oxidation states, from the all-ferrous to the all-ferric, but with different ligand. So there was a challenge to have the 5 redox state, but with the same ligand, and if possibly, the same thiolate time ligand to surrogate cysteine. And there is another challenge which concerns nitrogenase. It is a very complicated structure, double cubane structure with a carbide inside, and that's, I think it's a grail for chemists, and especially for you, for inorganic chemists. The work I will present you is not done in Marseille by women, but by men in Zurich. It was the group of Victor Rougel with his PhD student, Liam Goldwal, who defended his PhD last spring. And they have used a ligand that has been previously published in the literature for iron sulfur chemistry by the group of Tatsumi, is this thiophenol ligand with substituted mesyl groups in the ortho position. And by adapting a different strategy, they indeed managed to synthesize all the 5 different oxidation states. And you can see here the Mossbauer spectra that I've recorded at 80K at low field. And I've indicated by the red dots the average central position of the spectrum, and you see a continuous shift to lower velocity value. They are reported here as red dot on the graph on the right, and this is the same behavior as that observed in protein. But I would like to spend some more time on the 2+ cubane. They indeed managed to obtain 2 types of X-ray structure. One where the potassium ion that they have used is indeed encapsulated by the ligand that is on the left, and it is encapsulated by the mesyl groups of 2 adjacent ligands in the structure. And if they put a cryptant inside the solution, they chelate the potassium, and the cubane remains free of potassium. So if you compare these 2 systems, you may have access to second coordination sphere effect. They also manage, as they previously demonstrated by Tatsumi, that you can replace one thiolate ligand by a neutral tetramethylimidazole. And of course, you remain with a single potassium ion, and it can be either encapsulated or chelated by a cryptant. And once again, you may have access to the second coordination sphere effect. And if you compare with the system I've shown you on the previous slide, you may have access also to first coordination sphere effect. And before showing you the Mossbauer signature of these cubanes, as I've told you previously, they are made with 2 iron 2 and 2 iron 3, but they are fully delocalized. They are high spin and antiferromagnetic coupled, leading to a diamagnetic ground state. And in fact, it has been demonstrated more than 30 years ago that this diamagnetic ground state results from the antiferromagnetic coupling of 2 delocalized mixed valent pair, that is iron 3 and iron 2. But because the extra electron is fully delocalized between the 2 ions, you favor a parallel alignment of the spin, and that's double exchange interaction I worked when I was a PhD student. However, you may ask the question, you may see that, in fact, this mixed valence pair, you can locate them differently, either top or bottom, as on the left and right in the middle, and back and front on the right. And you may ask the question, are these systems equivalent or not? And to answer this question, I will present you the low-field Mossbauer spectra that we have recorded, either at 5K or at a slightly higher temperature, 80K. And you may see that the superimposition is almost perfect. You've got for the all thiolate and for the 1-imidazole system. You've got an asymmetric doublet, but more importantly, the line separation, that is the quadruple splitting, is temperature independent. This is I forget to say, sorry, that it was on the potassium-containing cubane. And if you turn to the potassium-free cubane, it's different. You've got now, at least at 6K, a symmetric doublet, and more importantly, now the separation is temperature dependent. And for this behavior, this means that you've got nearly degenerated electronic state, but presenting different orbital composition. So we assume that for the free cubane, where we have this temperature dependence, the free isomers are equivalent, whereas when you've got no temperature dependence of the quadruple splitting, the valence isomer are different, are not equivalent, and we may anticipate that because of the potassium that are chelated on two opposite faces of the cubane, you favor the isomer where the delocalized pair is facing the potassium, it behaves differently from the other two. However, the FT calculation didn't manage to discriminate between these two systems because the energy separation between the valence isomer is very small, whatever the system. But from the experiment, you have definitely a second coordination effect, and we suspect potassium encapsulation to lead to a differentiation of the cubane faces. We don't have any difference between the ultralight and the imidazole cubane, indicated that according to this specific property, we don't have any first coordination sphere effect. But if you turn to, if you think about biology, and that this type of cluster are involved in electron transfer, the fact that you prefer, you favor cubane faces, you may indeed induce a control of the electron transfer directionality. And the last example I want to show you is based on this imidazole cubane I've just shown you before. And as previously evidenced, once again by Tatsumi, it was possible to fuse two cubanes starting from this compound. And Victor and Liam managed to isolate a new double-stranded cubane structure. And you see that they are sharing this common sulfide. And the novelty is this sulfide here bridging two iron within the double cubane structure. And indeed, this structure is strongly reminiscent of what is observed in the nitrogenase K cluster. From the X-ray crystallography, if you look to the average iron-sulfur distances, you may see too short for the iron 1 and iron 2, and too long for the iron 3 and iron 4, indicating that on the top you've got 4 iron 3, and on the bottom you've got 4 iron 2. And it was indeed proven by Mossbauer spectroscopy, where at low temperature, low field, you have 4 lines of equal intensity, and the only way to pair them is the one shown on the screen, where you have an isomer shift value of 0.36 for the iron 3, and a larger one, 0.70 for the iron 2. And if you make the average, you are close to what we observed in 4 iron 4 sulfur 2+ clusters. You may compare this one of the subcubane with the one observed in the famous FMOCO cofactor of nitrogenase, which is the cluster responsible for the reduction of dinitrogen into ammonia. You've got the same oxidation state that is formally 2 iron 3 and 2 iron 2, but because the distances are different here in this synthetic cluster with inserted sulfide, the valence are trapped. And if you turn to a carbide inserted system, the distances are now similar, and that may induce, in fact, the fact that you've got delocalized pair in this system, and it may also be crucial for the catalytic properties. Well, I won't end my talk without thanking the person involved in the Mossbauer platform in Orsay, and especially Martin Clémencet, who make the spectra. The spectrometer running absolutely marvelously. I want to thank all the people in the team, Ricardo, which is also involved in Mossbauer spectroscopy, and all these people, all the persons that have been involved in the project I have shown you. And because I'm here because of a medal, I would like to thank two persons, Jean-Jacques Girard, who was my PhD director. He's in Orsay, and he helped me a lot. That's because of him that I've done EPR spectroscopy. And about 20 years ago now, Jean-Marc Latour, who was in my committee when I was employed at the CNRS, asked me to go to Grenoble to change spectroscopy and to turn to Mossbauer spectroscopy, which I came in Grenoble 20 years ago, and both of them always support me, encourage me, trust in me, maybe more than I do myself, and they always associated me for for their work, even if there is no spectroscopy, but because I'm also doing some computation, they associated me for specific problems. And because I'm a spectroscopist, I need collaborations, and it was also in chemistry or in biology, but I'm thankful to all these persons I've worked with, and of course, I will thank you for your kind attention. I hope to have shown you that indeed you can learn a lot from Mossbauer spectroscopy applied to iron. Thanks.
Thank you so much. So there is a time for questions. I'm very happy to see that the room is now completely full. That's a good look. And so if somebody has some questions or curiosity for Professor Blondet. Here we have a question by Rafa. Please.
Hi, thank you for your talk. Wonderful talk.
Thanks.
I'm a Mossbauer enthusiastic, not an expert. I like it a lot, but yeah, you know, the devices are not so common as an NMR on a UV device. I've done Mossbauer before in Erlangen and in Göttingen.
Yeah.
And always with molecular compounds. So for me, I'm impressed that you do that with proteins at so low concentration. So just curious, compared to a molecularly defined compound, how long does it take to take a typical Mossbauer?
So if you are, you may work on compounds, and the spectra I've shown you on all the synthetic iron sulfur clusters. It was in powder. They were not enriched in 57 isotope. It's the natural abundance. If you want to investigate them in solution, you should enrich in the 57 iron isotope. Otherwise, the concentration is too low. For a Mossbauer spectra, it's good to have 1 millimolar concentration in iron. For proteins, when you isolate proteins, what you do, you can't afford to work with natural abundance of iron. But the bacteria do the job. You put an iron salt in a minimal media where there is no more iron, so the only iron, or 85% of the iron that the bacteria has, is 57 isotope. So we are fully enriched when you investigate a protein that has been obtained like that, or cells. So it's not really a problem. The bacteria really do the job. And for the time to record the spectrum, you should be very patient. Few days for a protein, it may be a week. And it depends on the temperature, because you want the nuclei to absorb gamma rays. Well, it will move. It's like if you shot somebody with a gun, you will have a recoil movement. So you have to go down in temperature to increase the signal, to have a larger proportion, larger fraction of nuclei that are immobilized. Room temperature solution, you forget.
Yeah, I can see that in some of the signal to noise.
Yeah, it depends. It depends on several things, your concentration. And the main problem that we are facing now is the radioactive source, because it's a half-time of 9 months.
Yeah.
So when you have a brand new source, it goes quickly. For example, the last those spectra, they were recorded with a brand new source, and we did, in fact, 4 spectra within 3 days.
Oh, yeah.
But when you have a source that has 3 years old, it's more noisy and it takes longer.
Yeah, thank you.
Okay. Is there somebody? I have a curiosity about the fact that what is spectroscopy working on NMR spectroscopy, and I'm wondering if you was using a combination of the two spectroscopies let's say, NMR spectroscopy, Moss-Barrow spectroscopy, and maybe electron paramagnetic spectroscopy to get information of these clusters using, for example, selenium rather than sulfur for getting some NMR, special NMR information.
Well, for NMR, generally, you do it at room temperature and in solution.
Yeah.
So, and Moss-Barrow at room temperature and solution, it's not good. It's absolutely incompatible. So, obviously, you are working in different conditions. So, the comparison may be not so evident. But, of course, in synthetic cluster chemistry, they have also performed NMR in solution, and for the iron sulfur clusters, especially ORPR, I've shown you. Benedict Bura is collaborating to do Raman spectroscopy.
Okay, thank you. And let's make just a curiosity. I saw that you was mentioning the works by Professor Holm.
Yeah.
It was really a great pioneer in this chemistry.
Oh, yeah.
Really, let's say.
He had made too many things.
How far you are now with this, let's say, this related chemistry to the Holm clusters?
Well, I'm not okay. I never been a chemist mixing solutions on the lab bench. And,
was not a
but, yeah, but they are very good chemists, and I guess you are. So I think for the nitrogenase and its sorted cubane, it will come in nearly few years.
Yeah. Okay. So there is a question on the back, and then I think it's the last one. Okay.
Just about I guess. So my measurements are highly compatible with yours.
That's the game.
Yes. I was just asking how small of a coupling constant you can measure here, because I suppose that the coupling constants which you're seeing here are huge, because if you increase the field, you're still in the coupled regime.
Yeah, the cups we are using is about 1 centimeter diameter and a few millimeter eyes. For proteins, it's 400 microliter samples. But for materials, we may.
Sorry. I was referring to the coupling constant between the irons. The exchange coupling between the irons.
Yeah.
How large is that?
Sorry.
How large? What's the value?
You mean the coupling? Whatever you want. I don't.
Even very small. You can measure even very small couplings.
Yeah.
Okay.
Well, let me discuss.
One more question.
Hi, Marco Pino from University of Naples. I'm very happy to see you here. I will definitely bother you later on about some more questions, but now I would just like to know if you have ever worked with the heteronuclear iron sulfur clusters. If you have any, hint about this kind of stuff.
I've been working with Lucia Banchi, and that's all for the moment. Yeah. Because, well, in heteronuclear clusters, we will not probe the heterometallic site, but we will probe, of course, the iron nuclei. And so we have the response of all the iron will be sensitive to the magnetic structure of the whole system, but we will probe this specific iron sites. So, of course.
Just before ending, I forgot before to give you also the diploma, to account for. Thank you so much.
Buonasera, vi comunico che purtroppo la prossima lecture non si terrà perché Teodoro Laino ci ha appena comunicato che non può partecipare per un imprevisto. Per cui faremo il coffee break e voglio chiedere ai prossimi oratori di caricare le slide nel slide center. Vedo l'aula molto gremita, vi voglio anche comunicare che la sala A è collegata contemporaneamente per poter vedere le plenary e le keynote lecture, per cui se per esempio trovate lo spazio poco confortevole potete assistere alle plenary e alle keynote lecture nella sala A che si trova giù dove ci sarà la seconda sessione. Quindi ci vediamo puntualissimi alle 16:15. Ne approfittiamo per la passeggiata che il rettore ci ha consigliato di fare.
Prova audio. Prova. Ya kurre. Prova. 1, 2, 3, prova. Prova audio, prova. Si sente fatifette? Scracchia. Prova. Ah, boh, tutto su tu. Prova, 1, 2, 3, prova. Oh, vabbè. Riportato. Prova. Sì. No, ma quello è il problema, sono quando c'è la capitaneria di porto. Prova. Prova, test audio, prova, 1, 2, 3, prova. Mi metti la slide coffee break? Prova. Sì. Prova audio. Sì. Prova. Prova. 1, 2, 3. Prova. 1, 2, 3. Prova. Prova. Prova, test. test, test, prova, prova, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 1, 2, 3, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, prova test, prova audio, prova, prova, prova audio.
Prontos?
Good afternoon. I am Roberto Cipullo from the Department of Chemical Science of University of Naples. And let's start with the first session of this conference on sustainable catalysis. And to save time, I will begin with our first speaker, who is Francesca Arcudi, who will talk on photoredox cobalt catalysis toward pure ethylene. So please, Francesca.
All right. Good afternoon, everyone. Okay. It's really a pleasure to be here with you today, and well, let's say even more so in such a beautiful location. So thank you. And I'm very pleased to be kicking off this nice session and to share a bit of the work we are doing right now in our lab that is mainly about the production of pure ethylene. Now, before digging into specifics, or better, the angle from which we are trying to tackle this challenge, let's start with the why. Like, why do we care about it? And in particular, I said pure ethylene because, well, I don't have to explain to anyone how important ethylene is nowadays in industry, right? But what we usually don't consider is that we need this small molecule very pure. So let's take a step back. We produce ethylene in a very, very huge amount. We are talking about 200 million pounds every year. It's really a very important molecule in industry. We are producing a lot of pharmaceutics, but I think that it's mainly known for the production of plastics. Nowadays, we are producing something like 60% of all plastics, starting with this very small molecule. But the problem is that when we have to produce plastics, starting from ethylene, we need a gas feed that is very, very pure. And the problem is that the majority of ethylene that we produce nowadays in industry is based on the steam cracking. And this process, well established, of course, introduces some contaminants. And the most pernicious of these contaminants is acetylene. And it really is a problem because it poisons the Ziegler-Natta polymerization catalyst that we know about, right? And so what we do in industry to begin with is that we purify this ethylene gas feed. And specifically what we do in industry, we use a very consolidated procedure. We call this thermocatalytic process. It's very consolidated. As I said, it works very well. But especially in the last years, there is a growing concern about the sustainability of this process. Well, specifically to accomplish this conversion, that is the conversion of acetylene into ethylene, so it has to have a polymer grade ethylene feed, pure ethylene. We use palladium catalyst, so a noble metal catalyst that works pretty well, but it's prone to overhydrogenation. So the problem is that together with the conversion of acetylene to ethylene, we also end up having some methane. And the other problem is that these kind of catalysts work using a very high temperature and together with an external hydrogen gas feed. And another small problem is that we need a very selective catalyst, because don't forget that we are purifying an ethylene gas feed. That means we want to remove the impurity, like 1 volume% of acetylene, that is in our feed together with something like 20 or 30% of ethylene. That means we want to convert acetylene into ethylene, and at the same time, we don't want the ethylene to become methane. So this is the very, very important issue, right? So what we did back in 2022, it was to propose a photocatalytic strategy, an alternative to this well-established thermocatalysis. And we managed to basically fix these three problems at the same time using a cobalt catalyst in place of the noble metal, visible light in place of the high temperature, and water in place of the hydrogen gas feed. But of course, what we needed was a very highly selective catalyst, and we ended up with a cobalt catalyst that was able to reduce all the acetylene into ethylene with 99.9% selectivity in 28 hours. And here we are talking about industrially relevant conditions, meaning a very tiny amount of acetylene and a huge amount of ethylene just hanging around there. Now, specifically, what we proposed was to use this simple cobalt porphyrin catalyst, tetrasulfonato phenyl porphyrin, together with benchmark photosensitizer, ruthenium-3-PP, or even some more exotic photocatalyst, very common nowadays, some carbon nitride. And in general, what happened is that our reduced form of the photocatalyst is able to give us the low-valent cobalt-1 catalytic active species that do the work. Now, using this cobalt porphyrin catalyst, we also understood that the mechanism goes through the formation of a pi complex. And we were excited, of course, about the result, but we wanted to explore other opportunities in terms of mechanistics, possibilities to carry out this reaction. Still focusing on cobalt photoredox catalysis, what we found out later on is that it's more convenient instead to carry this reaction using a cobalt hydride intermediate, so the classic hydrogen atom transfer hydrogenation, in this case from acetylene to ethylene. And this gave us a polymer grade ethylene, so a polymer pure enough to be used directly in industry to produce polyethylene and other polymers. So the first work we did, actually is one of the latest, was performed by my fantastic postdoc, Anna, that is here in the audience. And what Anna found out is that cobaloxime is actually a very good candidate to perform this reaction going through this cobalt hydride intermediate. And as you can see from this graph, it represents basically the conversion of this 1 volume % of acetylene into ethylene. We managed to achieve this conversion in only 1 hour, compared to the 28 hours that under optimized conditions we got with our porphyrin. So this prompted us to investigate this even further, because it was clear for us at this point that a nitrogen atom transfer hydrogenation was indeed a good choice for this reaction. So we focused more on that, and currently, right now, in our lab, we are working on synthetic design of the catalyst, of course, and we are trying to improve and increase even more the level of complexity when it comes to our cobalt catalyst. Like in this case, of course, what we're trying to see is that an intramolecular proton relay can give us an advantage in terms of conversion. And I'm just showing some preliminary results, but indeed, this is what we expected was indeed what we got. Here in this graph, what you can see is the production of ethylene when we have our system under pure acetylene. And compared to the cobaloxime catalyst, that in 4 hours of irradiation of visible light got us something like 60 micromole of ethylene, using this Schiff base, cobalt Schiff base, we produce under the same condition, same visible light, and 4 hours of irradiation, something like 2.5-fold improvement in ethylene production. So just to say that I think that going towards this direction, so improving on the catalyst design, can be very useful also for this new kind of transformation. But as I try to highlight here at the bottom of the slide, an improvement in ethylene production can allow us eventually in the future also to substitute completely the way in which we produce ethylene. So eventually, if we keep improving on the ethylene production, we can replace the common steam cracking of hydrocarbon with the entire production of ethylene in industry, starting from acetylene, which can be, of course, very convenient because we can get a lot of acetylene from coal, for example. Okay, no, the ideal goal of everyone, a lot of people in the audience, is to produce ethylene starting from CO2, but as you see here in this very nice picture that Anna made, we are talking, in our eyes, about a medium-term solution. Yes, there are long-term solutions, very appealing, like the CO2 production, but in the meantime, we need to focus on alternatives to the steam cracking of hydrocarbons. All right, this was a small parenthesis. Now let's go back to the main topic, that is the purification of ethylene from acetylene. And actually, let's move a little bit to something different we are doing in our lab, that is, for this reaction, try to use an heterogeneous catalyst. Because homogeneous catalysis is wonderful, but heterogeneous catalysis can give us a lot of nice feature. In our case, we aimed at recyclability and the reusability of the catalyst, that in this context, right, for this specific reaction can be really of value. But in some of the works that I decided to talk to you about today, what we did was we built on the knowledge from the homogeneous system that we studied to construct some MOF catalyst where the linker are cobalt porphyrin or cobaloxyms. And this was very important for us because the reaction is still pretty new, even in terms of mechanistic understanding. So using this kind of linker gave us some kind of predictability. And also in this case, we managed to reduce all the acetylene inside a simulated industrial gas stream into ethylene with very nice selectivity. But most importantly, we were able to recycle and reuse this homogeneous cobalt catalyst for 5 or 6 cycles. It depends on the condition, of course. Some MOF are more stable than others. MOF are not perfect, we know that. But at least we got some degree of recycling and reusability of the catalyst. Now I'm going to stop somehow here because all my group is here at the conference, and so I'm going to leave to them with their poster the chance to talk about the ongoing work in our lab, which I think is the most exciting part of our research. Because up to now, I briefly try to talk about our systems, yes, our cobalt catalyst, but all these catalysts work very well in tandem with a photosensitizer. I mentioned the benchmark ruthenium tris-BP, and etc. But what the young researcher working with me right now are really doing is to optimize a single component photocatalyst, so as to have in a single unit the photosensitization and the catalytic function. And if we are good enough, and they are, not me, we can also have a unique platform that can be recycled and reused. I think this is, let's say, the end goal right now. So if you want to know more, please visit their poster. Anna is working a lot on cobalt single atom catalyst, as well with some homogeneous part that I showed to you before. And Brando is finally trying to switch a little bit from the cobalt photoredox catalysis, exploring other metals such as copper. And unfortunately, Sharon is not here, last minute illness, but she's also trying to explore colloidal photocatalysis for this nice transformation. Epilogue. I decided to put this as a conclusion slide. This summarizes the contribution from other research groups in this field. That's it. This is the state of the art. It is a very new reaction after our, after the first finding in 2022, other groups are of course working on this, and I just want to highlight that the majority of this work still deals with cobalt photoredox catalysis. So there is something special about cobalt, right? And this is the very last. May I just want to thank the wonderful researcher that decided to join my lab, and Stefano Agnoli and Laura Calvillo. Our lab is part of the big group that Stefano leads in Padova. And well, we might have, very soon, some open positions, especially for postdocs. So I will be around during the conference. Please stop by and we can chat a bit if you like this kind of topic. And thank you very much again for listening.
Thanks a lot, Francesca. The schedule is very tight, so maybe there is time for a very quick question.
Well, I don't know if it's quick, but let's try. So first of all, congratulations. This is really beautiful chemistry and impressive results. What is getting oxidized here? Do you get oxygen evolution? Do you get hydrogen peroxide? I mean, what is it?
So we usually like to work with a sacrificial donor. Not ideal, of course, but focusing more on the reduction side, up to now, this is what we do. But yes, ideally, we want to move there. It's a proof of concept, right? No, but with the scale I have in the lab, I'm not that presumptuous now.
Okay, sorry to interrupt, but there is time at poster session, so thanks again. I will go straight to the second speaker of this session, Andrea Piazzi from University of Bologna. He will speak on efficient ruthenium-catalyzed Gerber reaction for the upgrading of bioethanol to higher alcohols. And so, please.
Thank you for your kind introduction, and thanks for the organizing committee for letting me present our last result on the Gerber reaction project. So I will start with a bit of introduction on the bioethanol production and use. Nowadays, bioethanol is the most produced bio-derived compounds worldwide. Its production accounts for more than 110 billion liters every year. Main players of this production are USA and Brazil, which they produce bioethanol from the fermentation of first-generation biomasses, and they use it as a biofuel. Even though worse performances in terms of a fuel property are showed, and also some internal modification in endothermic engines are required in order to address the internal corrosion. So a lot of interest in the use of bioethanol as a renewable commodity has gathered a lot in the last few decades, thanks also to the plethora of compounds that we can make from it. And if you see in this plot, I compare the price of this product normalized on the bioethanol price with their global consumption. As you can see, most of the potential products are potentially profitable, including butanol. And the most efficient way to obtain butanol from bioethanol is by following the Gerber chemistry. Gerber reaction is a more than 100-century-old transformation is basically upgrading of an alcohol to a longer chain homologous. Is done by a three-step way mechanism. Firstly, we have the dehydrogenation of ethanol to form acetaldehyde. Then aldol condensation under basic condition occurs. And finally, the alpha-beta unsaturated aldehyde is converted to an hydrogenation step to the final alcohol. The same mechanism can be implemented for the combination of butanol and ethanol for the synthesis of C6 to C10 linear and branched alcohols. This alcoholic mixture can be used as it is, as a second generation biofuels, or each of these alcohols can be separated and used in a variety of different markers, such as cosmetics, surfactants, and plasticizers. The limitation of this reaction is nowadays that limits the scalability of this process are basically the Kaniz-Darren-Tischenko reaction, leading to the formation of sodium hydroxide and sodium acetate. So the approach in our lab is to work on the homogeneous catalysis for this reaction. And to do that, we developed the synthesis of two novel ruthenium catalysts bearing a non-innocent cyclopentadiene ligand. And in the ionic couple case, we have dimethylsodium salt as a counterion, or the same moiety attached in a carbene fashion to the ruthenium center. Both of them are water tolerant, and in the Kerbe condition, in couple with sodium hydroxide as a base, we are able to obtain a satisfactory conversion of ethanol with a higher selectivity towards butanol and a very good longer chain selectivity. They both work very, very similarly. And for the neutral complex, we recently published the DFT calculated mechanism. First of all, the pre-catalyst is activated by thermal CO release. Then the 16-electron catalyst is able to activate ethanol to form acetaldehyde, which undergo the aldol condensation step with the sodium hydroxide. Then the hydrogenated form of the catalyst is able to reduce the CC double bond and the polar bond, so the CO bond of the crotonaldehyde to form butanol. So as you can see, there are a lot of equilibrium involved, a lot of intermediates that are making the reaction not so trivial as a mechanism. Recently, we also demonstrate a very interesting trend using this time ruthenium ionic couple. As the volume occupancy in the reactor increases, so more ethanol is loaded in the reactor, a better carbon balance is obtained. So as you can see from the drastic decrease in C loss. So this is a very strong evidence towards the scalability of the process with our conditions. Therefore, we design a conceptual plant for the Gerbe chemistry in collaboration with the LEAP group in Politecnico di Milano. I won't go too much into detail, I've just summarized the part of the plant. First of all, we have the Gerbe reactor where the ethanol, sodium hydroxide, and the catalyst are loaded. Afterwards, we have a wall separation unit dedicated just for the sodium acetate separation. And finally, we have first the catalyst separation from the mixture. Then the ethanol is separated, the unconverted ethanol is separated from the alcoholic mixture and recycled back. And optionally, we have the distillation column to separate all the alcohols produced. Based on the engineering assessment, we also performed an LCA analysis in collaboration with the environmental chemistry group in Bologna. And here report just the carbon volume potential for different scenarios. The biobutanol production from the Gerbe reaction already is able to achieve a reduction in GWP compared to the standard butanol from fossil. Even bigger reduction can be achieved by using waste biomasses to ferment, to obtain bioethanol, sorry. And this was an approach that we already followed in a previous publication where we demonstrated that our ruthenium catalysts are are able to work even with second generation biomasses derived bioethanol provided by Caviro Distillerie. But what if we want to even more reduce the GWP of the reaction? For sure, we need to reduce our ruthenium usage, as well as the base consumption, but also, most importantly, to avoid the side production of sodium acetate, not only because it can lead to a better atom efficiency, but also because we can simplify the industrial plant. So following the experimental evidence that I showed you before, we began our journey into the scale-up of the reaction by working now with 5 milliliters instead of 0.5 milliliter of ethanol. And by using a pressure vessel, we are now able to work with just 0.02% of our flagship ruthenium NHC neutron complex, achieving similar performances, except for the increase in longer chain alcohol selectivities. And moreover, we also able now to work with just 5% more of sodium hydroxide instead of 20, without compromising too much the alcoholic yields. But since we were still producing sodium hydroxide, sodium acetate in this reaction, as demonstrated by the C-loss, we are trying now to shift to other bases, weaker, and to do that, we already screened 3 carbonates. The only one working is the cesium carbonate, apparently thanks to its solubility, but also potassium phosphate was found to be active, even though it's a bit lower in yields. We also screened, we did a close comparison with sodium hydroxide at different reaction times, achieving at every time we tested similar yields and selectivities. And this is also more readable by the plot, where I now compare the time distribution of all the alcohols production selectivity in time. As you can see, cesium carbonate is able to exceed the selectivity obtained with sodium hydroxide already at 2 hours, and also at longer times, but with a slight decrease in the selectivity. But most importantly, for the first time, we are able to obtain 0% of sodium acetate side production thanks to this new base, as confirmed by the HNMR analysis of the solid retrieved at the end of the reaction. So I want to give you some insights on the last work we are doing right now. We are now working at 30 milliliter scale, and to do that we reduced the catalytic loading to 0.01%, and in 5 milliliter scale we obtained the highest ton in literature with a new record for the TOF, 800, more than 800 H-1. And with 30 milliliters of ethanol, we are now facing a bit of issue on the yields, still a work in progress, but surprisingly, in the attempt to trap the co-produced water in order to push the selectivity furthermore to the alcohols, we obtain 0% of C-loss, so basically a 100% selectivity towards all the desired products. This was a very important result for us, and we will likely go on that later. So I'll give you some conclusions, some highlights of the work. We demonstrate the scalability of the process before with an engineering assessment and then with an LCA. We are now working on a higher scale with the cesium carbonate, demonstrating better efficiency and better selectivities. We also reached the top record for homogeneously catalyzed Gerber reaction by working with the mole sieves as a trapping water agents. In the future, we want to investigate more on the catalyst resting state in order to enable its scalability, and also explore a bit the carbonates in order to substitute the expensive cesium carbonate, and of course optimize the molecular sieve applications. So with that, I want to thank my lab fellows, my supervisor, Professor Mazzoni, all our collaborators, and of course you for your kind attention.
Andrea, we stick on time, and so now there is time for a couple of questions.
It's a beautiful lecture. Just a curiosity, because you showed the performance of your catalyst in this CC coupling, but how the performance of your catalyst compare with what is already reported in the literature with other systems? It's important to know.
Yeah, you're right. Basically, the performances are very similar. In literature, they all achieve around 50% of conversion, but most importantly, data is the selectivity. Selectivity can be vary a lot in base of you consider C loss or not. In literature, they don't consider C loss a lot. We are the only ones reporting it, actually. I think that basically all the ruthenium catalysts that they are published, they all reach the same amount of butanol. We are now focusing a lot on the longer chain alcohol selectivity as well. I think that now the challenge is the base and the sodium acetate formation, because basically they all act very similarly.
We are seems on time, so thanks again, Andrea.
Thank you.
And next speaker, Caterina Damiano from University of Milano, who will talk about sustainable carbene and nitrogen transfer reaction promoted by heterogeneous metal porphyrin complexes. Please.
Thank you for the presentation. Good afternoon. Before to start my presentation, I want to thank the organizing committee for giving me the opportunity to be here today and share with you our recent results about the use of heterogenized iron porphyrins for catalytic applications. As all of you know, to design a sustainable catalytic procedure, we should develop synthetic strategies that avoid the use of toxic and hazardous reagents, employing safe and, if it's possible, naturally derived compounds, combined with active and selective catalytic system that can be recovered and reused. In this context, an interesting class of iron complexes can be employed, in particular the one related to the heme group, one iron 2 porphyrin contained in a protein, the hemoglobin, responsible for the oxygen transport. The advantage in the use of heme is for the catalytic activity of this iron porphyrin and also the biocompatibility. In addition, the heme can be simply extracted from the protein by simply treating the hemoglobin with HCl. During this extraction, the heme is directly converted into the hemine, the corresponding iron 3 complex that show the chloride as axial ligand. However, even if this complex can be employed for catalytic applications, very few examples are reported of this iron compound under homogeneous condition. And the main problem is related to the very low solubility of hemine in most common organic solvents. For this reason, an interesting approach could be the heterogenization, with which we can anchor homogeneous species onto solid support by covalent or non-covalent bonds. In this way, it's possible to obtain a recoverable and a reusable catalytic system. So we thought about the possibility to anchor the hemine onto the Merrifield resin, a well-known resin employed for peptide synthesis and flow chemistry. We anchored the hemine bicovalent bond onto the Merrifield resin by using the strategies reported here, in which the chloride present onto the surface was firstly replaced by the azide, after the azide was reduced to the corresponding amine, and the amines were employed to form the amidic bond with the 2 carboxylic acids of the amine. During the last step, we performed a reaction in presence of methanol in order to replace the chloride with the methoxy ligand, in view of the very good effect of methoxy ligand onto the activity of iron porphyrins. So we characterized the material and we observed an homogeneous distribution of round-shaped particles. We also observed that this material is stable and the iron is present onto the surface. The complete characterization of the material is still in progress, in particular for the quantification of the iron. After the characterization, we then moved on to the catalytic activity, and we choose as target reaction the carbene transfer from the azo reagent to styrenes. As you can see from the general scheme, this reaction shows an high atom economy, and the molecular nitrogen is the only one byproduct. And it's a reaction well catalyzed by iron porphyrins. However, there is a problem in this reaction, and it's related to the explosive nature of the azo compound, in particular when low molecular weight diazomethane derivatives are employed. But to solve this issue, some years ago, Bill Morandi and René Koenigs developed the on-water and biphasic cyclopropanation reaction by the in situ generation of the azo compound. This reaction takes place by reacting an ammonium salt with sodium nitrite, and in presence of an iron porphyrin, the so-formed diazo compound can be converted into the product. So if we look at the general mechanism, according to the literature, the presence of water is required because we need to form the nitrosonium ion, and this ion is responsible for the formation of the diazonium salt that can be converted into the corresponding diazo compound, and this diazo compound, in presence of an iron porphyrin, can form the iron carbene, from which the carbene fragment can be transferred to this organic substrate. So, thinking about this mechanism, we at the beginning studied the reaction from the general point of view, and according to the literature, we observed that the slow addition of the sodium nitride is required to avoid the accumulation of explosive reagents. And also, even if it's known that this reaction can work under acidic condition, we finally found that the best condition was the one in which the pH is fixed by a buffer acetate. So, under this condition, we did some preliminary tests by employing the iron methoxy protoporphyrin 9 under homogeneous condition. We observed that the activity is more or less the same reported in the literature for other porphyrins, but the data can't be reproduced well because the material is not soluble in our medium. For this reason, we moved directly to the heterogeneous system in which the porphyrin is anchored onto the Merrifield resin. We did some preliminary tests by reacting styrene and alpha-methylstyrene with 4 different diazo precursors, one that contained the nitrile group, the trifluoromethyl-1, the ethyl acetate, and the chloride. And from this reaction, we observed that if there is the chloride, no reaction can occur. But with the other 3 precursors, it's possible to obtain the corresponding cyclopropanes from moderate to very good yields. And in particular, we observed that an higher amount of diazo precursor is required to obtain the final product in good yield. We optimized the reaction condition, and we did a substrate scope. The first one by using the nitrile-containing precursor with different styrenes, and we obtained the final product in yields up to 82% and the trans-cis ratio up to 83:17. We observed that there is a dependence from the steric hindrance. In fact, lower yields were obtained when the methyl group is in the ortho position or there is a phenyl group in the alpha position of styrene. But on the same time, it seems that the reaction is independent from the electronic properties of the substituent. As you can see, for example, comparing the reaction yield of paramethyl, parafluoride, chloride, and the methoxy substituents. At this point, we replaced the nitrile group with a CF3. We observed the same trend, but a lower yield, up to 53%. And replacing the CF3 with an ethyl acetate, an additional reduction of the reaction yield was observed, up to 36%. So, comparing all this data, we can conclude that the diastereoselectivities obtained are comparable to those reported for our previous system. And there is a strong dependence from the nature of the diazo precursor. And to explain this behavior and the entire mechanism, we are now performing some DFT calculation in collaboration with Dr. Gabriele Manca from the CNR of Bari. At this point, we tested the recyclability of our system, and we observed that the amine, the iron methoxy amine anchored onto the Merrifield resin can be recycled for 3 consecutive times with a very low decrease of reaction yield between the first and the second cycle, and also the material is stable. In addition to this, if we look at the entire process, it could be considered a sustainable procedure in view of the possibility to recover and reuse the catalyst, but also for the workup, because a simple filtration and extraction are required to obtain the final product. In addition to this, in view of the nature of the Merrifield resin, we are now performing some studies under floor condition in collaboration with Maurizio Benaglia from the University of Milano, and we observed that it's possible to obtain the product under flow condition in yields up to 33%. Anyway, this system should be optimized, and it will be studied in the future. After this, we thought, in addition to the possibility to additionally improve the sustainability of our system. So we thought about the replacement of the Merrifield resin with a more compatible solid support. And in particular, we employed color catcher, a cellulose-based material that was already prepared in combination with porphyrin to have active catalyst, sensor, and photocatalyst. So we anchored the iron methoxy protoporphyrin 9 onto the color catcher by simply dipping the color catcher in a solution containing the amine, and by treating the final material in methanol, again to exchange the chloride with the methoxy. We characterized the material, resulting stable and homogeneous, and we tested it in the model reaction between styrene and aminoacetonitrile reagent. We observed more or less the same behavior observed with the Merrifield resin, and in addition, we observed that this material is stable even if the reaction is under acidic condition. So we optimized the reaction condition, and we performed a substrate scope. We observed the same trend observed for the Merrifield resin, and the final products were obtained in yields up to 70%, and the trans-cis ratio up to 90:10. In addition to this, we also tried to scale up the reaction, and we finally obtained the product in 50% yield and the trans-cis ratio of 8:2 by using a very cheap reactor that is the simple tea infuser that you can buy at the supermarket. So, in conclusion, we described the use of heterogeneized biological iron 3 protoporphyrin 9, and we prepared 2 different materials. One in which the amine derivative was anchored onto the Merrifield resin by covalent bonds, and another one in which the same porphyrin was anchored onto the color catcher by ionic bonds. Both materials were able to catalyze the cyclopropanation reaction, and in addition, they represent the first example of heterogeneous catalyst active in the sustainable synthesis of cyclopropanes in water media. So, just to conclude, I want finally to thank all the people of our research group, all the founding sources, and of course, all of you for the kind attention.
Thanks, Caterina, and there is time for questions.
Okay, very nice talk, Caterina. Yep. And one question is, how did you quantify the catalyst loading, especially on the color catcher system?
At the beginning, sorry.
And regarding the covalent bond, how did you measure the catalyst?
Okay, at the beginning, the method it's the same, and it's the UV visible technique. because we can acquire the concentration at the beginning of the reaction, at the end, and all the amount of porphyrin in the washes. And after, for the color catcher, we already did the ICP, so we know exactly the amount of iron, as a consequence, the amount of catalyst, and we have to do the same for the Merrifield resin because we didn't do it now.
Just a curiosity, which kind of Merrifield design did you use? Because you know there are different kind of design that could be different swelling properties in different solvent, so that could help you in finding also a different behavior. Because
I want to try into different types. Yeah, but now for the moment I tested just the 100-200 mesh with the range of functionalization, so the chloride it's between 1.2 and 0.8 millimole per grams. I choose this type of resin because it's the one that were already employed in the group of Professor Benaglia for flow chemistry. So just to understand if the same type of solid support could be employed under the same condition. But I want to change the type because changing the type change the size of the sphere, so the functionalization, so everything. Maybe there is a resin better than this one. We don't know. Yeah.
polystyrene design and other kinds, because can change the aesthetic of the material.
Yeah, this is just the first one.
Okay. Thank you.
You're welcome.
Are there any questions? Okay, if not, thanks again, Caterina. And the next speaker is Marina Lamberti from University of Salerno, who will report on the N-heterocyclic carbon, zinc, and magnesium complexes for the synthesis of renewable polymers.
Good afternoon to everybody. Thank you, Roberta, for the introduction. The environmental crisis shifted the attention of polymer community towards more sustainable polymers. Aliphatic polycarbonates and polyesters fully fit to this category. In fact, they can be obtained from renewable monomers thanks to their biocompatibility and also to their physical and mechanical properties. They can be used in different fields, from packaging to mechanical and pharmaceutical uses. Moreover, thanks to their high oxygen content, they are biodegradable, and also they can be chemically recycled to the starting monomers or even to other molecules of interest. The ring opening polymerization of cyclic esters and cyclic carbonates performed by metal catalysts is a very efficient way to get both classes of polymers with high efficiency even under mild reaction conditions. With the good control of the polymerization process, both in terms of molecular masses and groups. We focused our attention on N-heterocyclic carbines because they have a high degree of modularity. In fact, it's possible to change the substituents on the backbone, also on the nitrogen atoms, and at the same time, it's possible to change the chemical nature of the backbone, thus resulting in good ligands for several metals. We choose zinc and magnesium because they are inexpensive, biocompatible, and also not harmful for the environment. Our first synthesis was the synthesis of this carbene chloride salt, which was already reported in the literature. The synthesis started from the reaction, the cross-coupling reaction of the first diamine with the 2-phenyl on the backbone and bromine toluene. The resulting product was condensed with the 3-ethyl orthoformiate, giving the chloride salt, which by reaction with the zinc precursors gave the desired complex bearing a carbene, an heterocyclic carbene, a chloride, and amido ligand. We also synthesized the magnesium complex with the same ligand, and also a zinc and magnesium complex with a second ligand, which differ from the first one for the stereochemical arrangement of the phenyl groups on the backbone. We observed by H1 NMR and also by DOSI that in the case of the magnesium complexes, the amine which is generated in the last step of the reaction remain bounded to the metal. Anyway, when we accumulated the DOSI NMR in a coordinating solvent like THF, the amine was depleted, as we can observe by the second DOSI. We also synthesized some cyclic carbonates following a green procedure reported in the literature by Bouchard by reacting CO2 with some diols deriving from carbohydrates. We got 1-methyl trimethylene carbonate and 2,2-dimethyl trimethylene carbonate with good yields, while we bought trimethylene carbonate. We used our 4 catalysts for the ring opening polymerization of lactide, caprolactone, and the 3 cyclic carbonates that we seen before. In all cases, we got to the desired polymers. Studying the kinetic aspect of the reaction, we observed the first or pseudo-first order reaction with respect to the monomer in all cases. Moreover, we can observe like the orange complex is always faster than purple complex, and the green one is always faster than the blue one. And this means that complexes with the ligand L1 showed higher activity with respect to complex with the ligand L2. By GPC analysis, we also observed that a good agreement between theoretical and experimental masses was always observed. For the ROP of substituted cyclic carbonates, we used zinc and magnesium complex with the first ligand, and we observed a slight higher activity with magnesium in the first case, and while with the methyl trimethylene carbonate, we got a higher activity with the zinc complex. Moreover, the second monomer is asymmetrical, so we studied also the regioselectivity in the synthesis of polymer with both catalysts, and in both cases we obtained a good degree of regioselectivity. We checked the end group fidelity, which was good because changing the alcohol by NMR, protonic NMR, and by MALDI, we observed the expected end group, and we also got a telechellic polymer by using a diol like the benzene dimethanol as the alcohol. As for the solvent effect, we observed that in THF and DCM the activity was higher. Anyway, the zinc catalyst was able to polymerize trimethylene carbonate even in a greener condition like with methyl THF, or in neat conditions without solvent, and we got higher turnover frequencies in this case. We checked the versatility of our catalyst by trying the synthesis of some copolymers with the 2 different strategies. The first one was a macro initiation with PEG, a telechellic PEG. We polymerized trimethylene carbonate, obtaining the 3 block copolymers. And the second strategy was a sequential copolymerization. First, we formed a trimethylene telechellic polymer, and then we added the lactide, obtaining an A-B-A copolymers with B-soft block. In all cases, DOSI analysis, GPC, and also DSC analysis confirmed the obtainment of desired copolymers. Part of our study was also devoted to mechanistic studies, as we or added an alcohol in the ring opening polymerization, we studied the reaction of the complex magnesium and zinc complexes with an alcohol in different deuterated solvents. In all cases, we observed the formation of the alcoholic adduct of the carbene. When to this mixture formed with the alcoholic adduct and the methyl species, we added the trimethylene carbonate or lactide. We observed the formation of a new species that we attributed to a carbonic adduct bearing the growing chain. When we added alcohol again, we got again the alcoholic adduct. We also prepared the alcoholic adduct on purpose, and by reacting the chloride salt with the potassium hydride and then with the alcohol, and we observed this was not active in the ring opening polymerization unless a methyl precursors was added to the mixture. So we proposed the mechanism already reported in the literature, which was a dual mechanism in which the methyl species acts as a Lewis acid activating the monomer, while the carbene, the free carbene, which is produced in situ, can act both as a nucleophile according to pathway A, attaching the monomer, either as a base reacting with the alcohol. In both cases, a linear chain is formed with the alcohol as the end group. As we observed that complexes with the ligand L1 were more active than complexes with the ligand L2, we looked at the chemical shift of the carbonic carbon bonded to the metal, and we observed the higher value for the complex zinc complex with the ligand L1. And it's reported in the literature that this indicates a higher electron donor ability of the ligand to the metal. So we thought to synthesize a new carbene and the corresponding zinc complex with a cycloacyl backbone. In this case, the chemical shift was higher, 205. And at the same time, this complex in the ring opening polymerization of raclactide showed an activity 2 orders of magnitude higher than the first zinc complex. Finally, we tested the performance of our zinc complex in the chemical recycling of PLLA by methanolysis reaction, both in DCM and in neat methanol. In the first case, we observed before the formation of oligomers and then the formation of methyl lactate, while in the second case, so in neat methanol, methyl lactate was the only product suggesting an unzipping mechanism of the polymeric chain. In any case, for both complexes, the performance were better under neat condition. In conclusion, we synthesized these zinc and magnesium complexes with NHC ligands, and they were used for the polymerization of monomers from renewable resources. We observed that zinc complexes show a higher activity with respect to magnesium for lactide and methyl TMC, while magnesium complexes were more active than zinc for caprolactam, trimethylene carbonate, and dimethyl trimethylene carbonate. We tried to explain this observation thinking to steric effects, because for the first two monomers we have a methyl substituent closer to the active center of the monomer, but we are trying to study better this difference, and also some colleagues of us are trying to study this from a computational point of view. We also observed the formation of the alcoholic adduct of the carbene in situ, and so we supposed that the The carbene works in synergy with the emitter species according to a dual mechanism. Finally, we showed that our classes of complexes was able to close the life cycle of PLLA by methanolysis reaction, so forming methyl lactate, which can be used as a green solvent or can be used to get the monomer back. I would like to thank Federica Tofano, the PhD student, and Federica Santulli, the postdoc student who worked on this project, Prof. Grisi and Prof. Mazzeo, my colleagues I collaborated with, of course, all the guys in the lab, University of Salerno and the Italian Ministry of University for funding, and the organizing committee for giving me the opportunity to talk about my work, and of course, all of you for your kind attention.
Hi, thank you. Very interesting, very good. Very impressed because the chemistry of magnesium and zinc coordination chemistry is not easy and not that intuitive as with other metals, where you see color change or you have distinctive spectroscopic signatures like vanadium in EPR or rhodium in NMR. I have kind of two questions. First, I imagine you did the corresponding control experiments in the polymerization studies using only zinc or only the ligand in order to see if the polymerization is simply anionic polymerization triggered by the base or cationic polymerization triggered by the metal center. Second, I'm wondering about the stability of this species in alcoholic media, because magnesium and zinc are very oxophilic. So I don't know if you also study that. Thank you.
Okay, as for the first question, actually this carbene ligands was not, we were not able to make the free carbene ligands, so we could not try the blank just with the ligands. We did some experiments, an additional slide, because we were of course curious about also the effect of the additional amine, so we tried, so we used it's this one. We tried the kinetic study of both the polymerization mixture that we used, and also by using the alcohol adduct in the second part of the slide, we used the alcohol adduct with the mixture of the chloride of the metal and the amide of the ligand, just to see if it's formed the same species that we supposed, which was a metal species mixed with the chloride and amido ligand. And actually the activity was not exactly the same, but the order of activity was the same. So this was our check. And also we tried the metal amido alone, and in that case the activity was higher, but without control of the polymerization process. The second question was, sorry? Okay. When we tried the reaction of the complexes with the alcohol in deuterated solvents, sometimes we used an excess of alcohol, and what we observed was that the alcoholic adduct was formed, but this was stable. So we think that the alcoholic adduct of the carbene is formed, and that one is the one that generates the carbene. It reacts with the water. With water, you have another product. So it's not stable in the presence of water. Thank you.
Okay, let's thank Marina again.
Thank you.
The next speaker is Tala Munir. Sustainable innovation chemical synthesis and catalytic transformations.
Hello and thank you for this introduction. As you have seen my topic, it is sustainable innovations. I will represent mechanochemistry as a sustainable innovations, and you can see the highlights. It is actually two parts of my presentation. First, I will focus on mechanochemical methodology. That is actually a comparison of solution-based methodologies and mechanochemical-based methodologies. And in the second part, I will try to discuss some results in comparison with solution-based approach and mechanochemical-based approach. So, as we have very much familiar with organometallic synthesis, generally it is a multi-step procedure. We have to choose a particular precursor, that is, we are very much specific with the precursor, and also with the solvent and type of solvents. With different procedure, we have to degas the solvent, provide some inert atmosphere in the system, and also we have to follow up our work with some improvements in the system, purify the product, and also drying of the product, isolation of the products. And considering these systems, we have the goal that we have to convert reactants into products. But for that, we have to provide a medium of reaction in a conventional base approach, and also we have to do the follow-up steps, including vacuum, washing, refluxing, and drying of the systems. But if we observe as a chemist or as a scientist, we have also some environmental impacts that are involved in these systems. Like there are, for example, if we are using too much solvents in the system, for sure there is solvent waste. We are consuming too much energy in the system. There are also procedural factors that increase the time of reaction of a system. Also, there are impacts on the reaction that we observe in the form of several intermediates and byproducts in the system. And if you see this picture, it looks absolutely beautiful to us. Like we are chemists, so we enjoy these things. Like it is a very, very, very enthusiastic. This makes us proud, like we are a genius. But if you see, the purpose for us is to make things easy for others. Researchers are to make the things easy for others, not to make it more complex. So we offer, So we offer another alternative, which is probably proposed my supervisor in the previous talk in also this conference 2023, by offering mechanochemistry as an alternative to solution-based chemistry. And we have a lot of pros and cons. So mechanochemistry is not new. It is present from the ages. You remember mortar and pestle, we started by the Stone Age. It is also highlighted at top 10 innovative technologies by IOPEC. And also, if we see in the results from the upcoming slides, there are also some products that are actually not available or cannot be prepared by solution-based methodologies, but they are very much feasible and isolatable in mechanochemical synthesis. And mechanochemistry, if you take a comparative view, mechanochemistry for sure is a solvent-free, or you put a very small amount of solvents in the system. Reactions are pretty much fast. We will see some results. And also there is no purification steps, or very low purification steps are present. And also for sure it is green and sustainable innovation. If you compare this one with solution chemistry, we use a bulk of solvents. We know that we generally don't measure the solvent. We just put in the slang just to increase the contact of the system. And also the conditions are pretty much intense. We have to do comprehensive workup, and also there are very much environmental effects that are present in the system. So we offer a very simple, very, very clean methodology rather than the solution-based approach. We just take the reactants, put them in the milling container, we mill them for appropriate time, and the products are there. You just do the characterization and your products is there. By applying this methodology, we have, what happens inside a jar is still sort of a mystery, because there are very few approaches. The main approaches that are there is two models that say there is a formation of hotspots that are actually high energy states that are main reason for conversion of reactants into the products. And if you see in a lower diagram there, we have different type of mills. This is a vibratory mill, a rotational mill, and also type of extruder that are used for synthesizing the products. So this is the first paper that we have published on mechanochemistry, and we use these type of precursors, a different precursor, and the ligand is piperidine. So we tried piperidine with all these ligands, and we were able to isolate more than 10 compounds, piperidine metallic precursor with pretty high yields. And also the main focus of the paper is the comparison of solution based approach and mechanochemical based approach, and the results are actually very much inspiring, that we can see the comparison of yield and time of solution based approach and mechanochemical based approach. If you see the blue bars in the upper in the time graph, you can see this one for the complex one, that is the complex of parasamine, ruthenium, piperidine complex. The time for mechanochemical synthesis is very much low, that is just 10 minutes as compared to the time that is more than 3 hours in solution synthesis. And if you compare the same in case of yield, the yield comparison you can see in the red bars, it is around 70%, and you can see in case of mechanochemical synthesis, it is the reaction is quantitative, 100% yield is isolated. And if you compare the green parameters, the numbers don't lie, you know. You can compare the E factor of our methodology and E factor of solution based approach. There is very much a difference, very much high difference. You can see E factor of most of our work is almost 0, and E factor of solution based synthesis is very much high. And also if you compare the effective mass yield of mechanochemical synthesis that we have done and solution based synthesis, effective mass value of our work is more or less optimistic. Ideal case, 100% yield is observed. And also we then shifted our work to other compounds that are synthesis of ruthenium, PP, NN complexes. And for this approach also we applied one-pot approach as compared to the classical system of ruthenium, PP, NN complexes, where it is a multi-step procedure. This is actually the procedure that is usually employed in the literature. We replace this procedure with 2 other approaches. First methodology that we use is starting from ruthenium trichloride to ruthenium cod, and then from ruthenium cod to ruthenium PPNN complexes. The other methodology that we applied is directly from ruthenium trichloride to ruthenium PPNN complexes, and with both of these methodologies we have synthesized some interesting complexes. We will see in the next slides. Okay, for the first route that we propose, that is based on ruthenium cod, we chose these dinitrogen ligands and these diphosphine ligands with ruthenium cod, and with these options we have synthesized more than 15 complexes that are ruthenium PPNN complexes. And you can see a series of DPPFA complexes with the piperidine, phenanthroline, and also we have used diphenylethylene diamine. Of course I have to manage the slide, so there are some complexes that are not present here. Just an interesting point here, you can see there is a DPPE in the system. If you in the literature they usually report that DPPE, whenever they try to synthesize these type of complexes, there is a formation of homoleptic species because of the bite angle of the ligand and other problems. But here we also observe the homoleptic species, but altering the conditions of the system, we were able to isolate these complexes in pretty much high yield. Moving to the other route that we have is directly from ruthenium trichloride to ruthenium PPNN complexes, and for sure we have to reduce ruthenium 3 to ruthenium 2, which was a challenging task. But if you study the literature, the work of G. Wilkinson, he actually used double excess of phosphines to reduce the ruthenium 3 to ruthenium 2, which says that phosphorus or phosphines or diphosphines can act as a reducing agent. So we changed the stoichiometric designs. We added 25% excess of diphosphines just to make the reaction more stoichiometric, and we changed the system to for the isolation of the product, and the confirmation was done for the filtration. We confirmed that there is no ruthenium trichloride free present in the system. And there are a lot of application of these complexes. They are very much famous for transfer hydrogenation reactions and reduction reactions, water oxidation and other type of reactions. Also, we applied these complexes not for transfer hydrogenation reactions, but for oxidation reactions. In the literature, they have never been tried for oxidation reactions. But here we chose a secondary alcohol, and we chose a ruthenium catalyst. For sure, we need an oxidant in the system, and we chose these catalysts, and you can see that time is pretty much low as compared to the system that are present in the conditions that are present in the literature, because in the literature the oxidation of secondary alcohols is considered to be quite tough, and we were able to isolate the product in very much high conversions. And if you see the advantages, for sure there are no solvents, there is low energy consumption, and also you can scale this reaction. The approach is quite sustainable, and for sure it is a green methodology. Also, we tried some other catalysts. For example, this catalyst was prepared by our previous work with piperidine, and it gives us 80% conversion. This is another catalyst that we have in the lab that was also synthesized by mechanochemical approach. When we try to add piperidine in the same catalyst, we increase the conversion from 40% to 77%. And also the best catalyst is with the acetates that we have, a type of ruthenium PPNN complexes that gives us 98% conversion. So in the summary, we have seen till now good results, pretty much interesting results from ball milling and mechanochemical synthesis. For sure, it is a green synthetic route. There is no or very low in microliters amount of solvents are used, and also E factors and effectiveness yields are very much promising. We have synthesized metal piperidine complexes. That work is published, and now we are almost ready to publish another work of ruthenium PPNN complexes. The results are more or less ready, and also we are now working on mechanocatalysis for the oxidation of alcohols. For sure, there are some limitations with the mechanochemical approach, that there are some cases that isolation of the product is problematic in some cases, and we also see decomposition because of high or intense milling of the system. We have a lot of milling to do. We have a lot of plans ahead, and we assume that mechanochemistry or ball milling in organometallic synthesis is a good approach, and we are expanding it in various manners. And that's all. Thank you so much for your kind attention. I would like to thank my supervisor, Professor Daniele Zucaccia, and also Professor Walter Barata, Professor Eleonora, and my other lab mates, Fabio and Leonardo. Thank you all for your kind attention.
Time for just one quick question.
Yeah, really very interesting approach. Thank you for this talk. It's just a curiosity, but maybe I missed something. But if you go back to slide 9, I think, is when you start from ruthenium 3, all right, you're getting a bisphosphine oxide, right? So I don't see oxygen. Well, there is water in the ruthenium chloride, right? So it may be that there is it's in the system, and so you're probably getting HCl that evolves. Have you?
We have water with ruthenium trichloride. We have water with ruthenium trichloride here, but the purpose is just to reduce ruthenium 3 to ruthenium 2. So we need electron transfer. So if your question is the oxygen is present inside the jar, because we are not doing in an inert environment.
All right. The curiosity was just if the phosphine is able to abstract chlorine directly to make a chlorophosphonium.
We didn't observe the.
Thank you.
Thanks a lot. Thanks a lot.
Okay. Thanks, Tala. And next speaker is Christian Silvio Pomelli from University of Pisa, who will talk on computational experimental study on inexpensive of green ion liquid catalyst for CO2 activation.
Thank you. I start, and I think everybody in the audience know there's a lot of research about silicon carbonate for carbon dioxide fixation, are useful solvents in different application, additives for gasoline and fuels, preparation of polymers, etc. And the principal synthetic route is from epoxides and for a ring opening reaction, and the most accredited reaction mechanism is the need of a positive and a negative catalyst, a nucleophile and normally a cation, to activate both silicon oxide and carbon dioxide. And there are a lot of catalysts, a lot of bivalent catalysts with both the features on same molecules, a lot of very complex molecules. We propose a very simple catalyst based on the mainstream ligand for metal ions, ethylene, tetracetic, diaminic, etc., and using different bivalent cations, very not toxic, not expensive, and using as a counter ions the main cation of ionic links is butyl, methyl, imidazolium. You can see the synthesis is very simple. You use water, methanol, methyl carbonates are the standard template for ionic liquid synthesis, anionic exchange, halogen-free anionic exchange. And this is very simple. This catalyst is very soluble in epichlorohydrin and a lot of another oxyranes, and we can operate in a synthesis-free, in a solvent-free synthesis, and is a good solubility in reaction media. We can easily recover the catalyst. We have nucleophile carboxylate and cation the same molecules, and the two cations are not here in the scheme, are stabilized double charged complex. This is optimized structure of our catalyst. This is the one with magnesium, with the two cations can influence, stabilize what hydrogen bond, especially with the C2 acidic proton. And some experimental evidences, there are high yield of conversions for almost all the cations we have taken on consideration. There is a small problem, with copper, in which the catalyst reacts slowly and irreversibly, go to be degraded with epichlorohydrin. But in meantime, it can react with carbon dioxide. If you want more details on experimental part, you can look at poster 58 with my coworker Luca Guglielmiero. He present a poster with experimental approach with others epoxies, other type of complexes too. For the computational part, I was inspired from this paper. This is a very nice paper and present a methodology to accelerate the computation with using hybrid basis set. A basis set is the set of functions on which the wave function is optimized, and in this paper established two protocols, one low level, the one in green, and high level, the one in gray. In this work, I used the green, the protocol of low level. It's not too low level. This function is a double hybrid functional. I have also a part of MP2 energy inside. Is a good functional. And small basis sets on a portion of the system, and a larger basis set on the interesting portion of our system. And this is a simpler approach than onion multi-layer methods, and simply you can ask any density functional code to use some basis set on some atoms and some other basis set on other atoms. This paper validates the results against high-level calculation, and especially for organometallic reactions. This figure on the left is one of the examples in the paper in which the blue atoms, obviously the metal atom and other atoms, are high level, a larger basis set are the interesting part of a system, and black atoms have the smaller basis set. You can see there is not a problem to partition also small molecules into parts and save basis set in computational time. You can see that in our system, the reduction of number of basis functions almost the half in the model with respect to complete basis set. It is a big saving in computational costs. I use also continuum solvents where the reaction is in epichlorodyne with the electric constant of epichlorodyne, and this is a methodology I use here. I use ORCA on a free program, free for academics, and a 5-year-old computational server, which is not too expensive calculation nowadays. And this is detail of mechanism. In the first step, we have the activation of epoxide, one of the carboxylic groups. These carboxylic groups, this only is described by large basis set, and with metals activate the oxirane, stabilizing with 2 bonds. Now, and after enter the carbon dioxide form a carbonate group, react with oxygen, and then the product is obtained by a ring closure and formation of a product. In all the system, the 2 cations are around and stabilize the system. This is an example of transition state of the first step. You can see on the left the carboxyl, the opening oxirane, and the coordination bond with magnesium. And we can see, for example, this this C2 hydrogen of the imidazolium in some way stabilize the oxygen. The activation energy are large, but the reaction take place, and you remember there's a high concentration of epichlorohydrin is a solvent, and probably the mechanic ballistic interaction between the complex and the epichlorohydrin molecules, solvent molecules can furnish the reaction, the energy for this first step. There is not great difference between the ions. We can see that there is difference in stability of first step, intermediate, and the copper is more stable. Why the copper have this problem? This is only an hypothesis. With the exception of calcium, all other ions have almost the same size, are very similar size. And but the complex with copper show one of the carboxylides, not the one that stabilized the system, but one of other three, is a distance greater than another comparable ions. We can see there is some difference in geometry in the complex. Every atoms have a slight different geometry, and this can be a difference. In the copper, the chelate is slightly open. Carbon dioxide now enters the stage, and activation of carbon dioxide is, for geometrical point of view, the important is the bending of oxygen-carbon, oxygen angle. We can see in transition states of this phase, the angle of magnesium is the best performer in catalysis, in experimental reaction, is have an angle smaller than another one, and a very low barrier in this step, less than 10 kilojoules per mole. And the last step is the carbon, the ring closure, 5 ring closure, and the product. And for schematize all the reaction, we can look at two distance, two metal oxygen distance, with oxygen on the vertical axis, with the carboxyl on the horizontal axis, and all the system describe a cycle, because the first on the last system, there is a different molecule outside, but the complex is the same. And we see there is a lot of difference between the path of different ions, and there is also difference in activation energy, and every of the system have its different details, can be analyzed in this way or in another way. And this is all, and this is our website, the logo of our research group on ionic fluids, University of Pisa.
Okay, thank you, Krishna. Maybe we are a little behind schedule, so if you don't mind, I would postpone the discussion during the poster session.
Okay.
And I would go to the next speaker, Francesco Noscito from University of Bari. Highly dispersed nickel 0, cerium 3 catalyst over clay mormolonite K10 in low temperature CO2 methanation. Please.
Okay. Thank you, Chair, for the introduction. And I want to thank all
the committee for this opportunity. So the context in which this work is placed is to find new application or improvement about the carbon dioxide conversion in order to help the climate change mitigation due to the increasing of carbon dioxide concentration in the atmosphere. So we know that the carbon dioxide conversion can be divided mainly in three big categories. So-called at low energy processes in which carbon dioxide can be used as it is, in which carbon dioxide can be inserted into an organic molecule in which, for example, an activate oxygen or nitrogen or carbon is present in order to obtain carboxylic acid, organic carbonate, or carbamates. And also, we have other two kind of processes in which carbon dioxide calls high energy processes in which carbon dioxide needs to be reduced. Needs the reducing of the oxidation state of carbon. And in these two processes, molecular hydrogen or water needed as hydrogen source. So, one of the most important industrial process are the Sabatier reaction. Why this process is important? At the first, because with this reaction we can convert a waste, that is carbon dioxide, into an hydrocarbon. Water represents a byproduct of this reaction. This reaction is an exothermic reaction. And the fact that methane is the main product, there is an availability of the methane distribution network and also the market that is deeply known. In addition, we can use this reaction in other industrial fields. For example, to valorize the syngas obtained by pyrolysis of biomass, or in order to valorize the biogas produced after fermentation. But this reaction shows also some limits. For example, it needs a previously captured and purified carbon dioxide, needs previously produced hydrogen. 50% of this hydrogen is lost to produce water, and depending on the reaction conditions, the reverse water gas shift reaction can subtract hydrogen to the Sabatier reaction. So, for this reason, catalysts represent a key aspect in order to increase the efficiency of the process and reduce the overall costs. And the catalyst design represents the pivotal role because we need to study an active metals able to activate hydrogen, to activate carbon dioxide, that shows specific acid-based properties also to absorb both hydrogen and carbon dioxide. A high surface area in order to better disperse the active metal to reduce the possibility of sintering, and also a specific support with low carbon affinity that reduce the possibility of pollution of the catalyst. In literature, I reported several examples of Sabatier reaction application, and in most of cases, noble metals are used. Ruthenium, rhodium, especially that effectively shows high activity and also high selectivity, but there is the problem relative to their high cost that makes not sustainable the process. And also, this kind of metal works better at very high temperature. So nickel for this reaction shows a very good compromise between activity, selectivity, and very low cost. For this reason, we studied a catalyst that contains nickel and in presence or absence of a promoter, comparing different kind of support in which nickel can be adsorbed. And in this context, I select only the main kind of support that we studied, all based on alumina silicate on alumina silicate moiety, that are the very low cost support. All this support shows very different textural and surface properties. For example, the titano-silicalite present an hollow structure. Hydrochalcite and montmorillonite present a layer structure, and gamma aluminum oxide a highly porous surface. Then hydrochalcite present an anionic exchanger properties, montmorillonite and cationic exchanger properties. And considering the properties we studied, for example, we can see that titanium-silicalite shows the higher pore volume, or MK10 the higher acidity, and hydrochalcite the higher basicity. So our purpose is to, it's not important, don't worry. Our purpose is to study how these properties can be used in order to well design the most active catalyst. So for the synthesis, we use the same approach, that is the strong electrostatic adsorption. At the first, to investigate the effective amount of nickel that can be strongly bonded on the support. Strongly, but not so strong, because after we see that the strength in which nickel is bonded on the support is very important. So also we compared how these properties influence the activity and the selectivity of the reaction. For example, we can see that higher is the acidity and higher is the amount of overall nickel that is absorbed. And if we observe the picture D, obviously can appear obvious that increasing the amount of active metal increase the activity of the catalyst, but this is not true, because if we use very high amount of nickel, we increase the possibility of sintering of nickel, or increase the possibility of pollution of the catalyst. In the picture B, we can see that conversely, the basicity contains increase the possibility of absorption of carbon dioxide. So this means that we need a precise compromise between acidity, basicity, surface area, and all the different characteristics of the catalyst. For this reason, the catalyst design needs several tests in order to exactly find the correct catalyst. In addition, how we can see from the XPS investigation, we have at least two different kinds of nickel oxide that can be formed on the surface of the catalyst. On the surface, also in the internal layer of the catalyst, because we observed that, for example, using titano-silicalite, nickel is positioned mainly in the holes. If we use hydrochalcite, mainly in the surface. Instead, if we use MK10, is homogeneously dispersed both in the surface and in the internal layer. But we observed two different kinds of nickel oxide. Nickel oxide is formed during the calcination process, because we start from a salt precursor of nickel, that is nickel nitrate. And during the calcination, we convert it into nickel oxide, and the last step of the synthesis of the catalyst is the reduction, because the active metal is the nickel at the 0 oxidation state. So if we observe the table of the calcined catalyst, we have a nickel oxide-like catalyst that's very similar to free nickel oxide, that is strongly bonded to the surface only for diffusion. But we have also a nickel aluminate and nickel silicate species that are formed during the calcination. And this different kind of nickel oxide has different behavior during the reduction process, instead increasing the energy bonding between the nickel oxide and the support, increase the temperature of reduction. That is a negative fact. Higher is the reduction temperature, higher are the costs to reduce the nickel. So, in order to increase the activity, we checked the effect of a promoter. Also in this case, we checked a different kind of a promoter, but cerium oxide and cerium-3 in particular shows a very good ability to uptake carbon dioxide, considering that on the surface of the cerium oxide there are several oxygen vacancies that are able to uptake carbon dioxide. But also in this case, we observed different behavior of this support. For example, in the case of hydrothalcite, we observed cerium in the internal layer and nickel on the external. On the alumina oxide, both are on the surface, obviously, because it only porous structure. But nickel cover almost exclusively cerium oxide. Instead, in MK10, the distribution is perfect. We obtained both nickel and cerium in internal and external surface. That means that is a very good dispersion of the promoter and of the metal active. And in this last slide, I report the activity of all these different catalysts, comparing the effect of cerium, of the promoter on the catalyst. And we can see that both nickel, cerium, alumina, and nickel, cerium, MK10 reach almost 80% of yield in methane, and both with 100% of selectivity. But what is the difference between these two catalysts? Is the long life of the catalyst. In the case of the nickel, cerium, alumina, after the second cycle, the activity decreases because a part of nickel is lost. And instead, in the case of nickel, cerium, MK10, at least for 5 cycles, the activity and the selectivity remain practically the same, without any reactivation of the nickel. So, in conclusion, with this work, we prepared a robust nickel, cerium, MK10 catalyst that permit to convert with very high yield and very high selectivity methane at very low temperature, that is 216 degrees. Generally, this reaction occurs at temperature higher at 350 degrees, and with low reaction time, that is 3 hours. And the time available is finished, so thank you for your attention, and I'm available for some questions.
Okay, thank you. Maybe just 20 seconds for a very quick question, otherwise we will post.
My collaborator is on poster 61, and I have to stay close to him. So if you want some questions.
Okay, the poster session. Okay.
Okay.
Thanks again. And we'll close this section with Linda Leona from University of Naples, who will report on design enzymes housing functional histidine brace copper binding sites. So, Linda.
Thank you. It's a pleasure for me to be here. Good afternoon to everybody. I will show you some of our work on artificial metalloenzymes hosting functional histidine brace copper binding site. The histidine brace is a coordination motif that involves an N-terminal histidine that binds a copper ion through the N-terminal amino group and its delta nitrogen of the imidazole side chain. Copper is usually further coordinated by an epsilon nitrogen of an internal histidine, and this kind of copper center is found in metalloenzymes called lytic polysaccharide monoxygenases, or LPMOs. The LPMOs are metalloenzymes that are gaining increasing interest over the past 10 years because of their ability to catalyze the breakdown of insoluble recalcitrant polysaccharides such as cellulose and chitin that are highly abundant in biomass. So they are finding applications in waste valorization. So, in particular, they catalyze the cleavage of glycosidic bonds in polysaccharides upon activation of molecular oxygen or hydrogen peroxide, so through an oxidative mechanism. Several artificial models of LPMOs have been developed over the past few years, either in the form of small molecule coordination complexes, metal binding peptides, or through the engineering of natural protein scaffolds. And here, the research group where I currently work in Naples has approached this challenge through a de novo protein design strategy. And we have developed two models of LPMOs called mini LPMO and the histidine brace. So the first model, mini LPMO, is based on a de novo designed scaffold called alpha-2D, which is a non-metal binding protein adopting a 4-helix bundle structure, in particular with an anti-bisecting topology. So our design started from the structure of alpha-2D, where we selected the best positions to insert the histidines, and then after reconstruction of the sequence, we obtained mini LPMO, which is actually a homodimer hosting two copper sites. So you can imagine that the dimerization is crucial for this kind of system, and we have investigated this behavior through both circular dichroism and analytical ultracentrifugation analysis, both in the absence and in the presence of copper-2 cations. We can see that despite minor structural changes, copper binding is crucial for mini LPMO to adopt the desired dimeric structure, while it behaves as a monomer in the apoform. So once assessed its copper binding ability, we have investigated the coordination properties of the copper site in mini LPMO, and we have employed both UV absorption spectroscopy and DPR spectroscopy over a wide range of pH values. So from the analysis of the visible spectra here on the left, we can see that moving from acidic to alkaline pH, we observed a blue shift of the DD absorption band of the copper center, concurrent with an increase in the extinction coefficient, which indicates that we're moving towards a less symmetric and a stronger ligand field surrounding the copper ion, which indicates the formation of a histidine brace binding site. Also, changes in the EPR spectra with the pH are consistent with those found in natural LPMOs. So we have an indication that actually at neutral to alkaline pH, mini LPMO holds a histidine brace copper binding site. Then, to assess the functionality of this copper center, we have studied a model reaction. And in particular, we have selected paranitrophenol glucopyranoside because it mimics the glycosidic bonds found in cellulose, but it offers the possibility to follow the reaction very easily through UV-Vis spectroscopy. So we used hydrogen peroxide, which is actually the preferred co-substrate by LPMOs, and explored this reaction at different pH values. We can see here that actually mini-LPMO is capable of cleaving the glycosidic bond in the model substrate, and it reaches the highest activity between pH 7 and pH 8.5, thus indicating the presence of a functional histidine brace in these conditions. But then we have moved forward and we have designed another model with the aim of improving the properties from a structural and catalytic point of view with respect to mini-LPMO. And we have designed DSB, which stands for Designed Histidine Brace. In particular, we have selected another de novo scaffold, still an alpha helical scaffold, but in this case, the starting protein is called coil-SER, which is a three-stranded alpha helical coil, a homotrimeric, and we have selected this scaffold because the longer helices compared to mini-LPMO would favor the dimerization even in the absence of copper ions. Further, we wanted to construct a platform for easy screening the effects of modifications at the histidine brace. So we have first constructed a loop for joining two of the three helices, giving rise to an HLH helix loop helix peptide, which recognizes a single helical peptide where the N-terminal histidine is introduced. So with this system, we have actually, and we are currently studying the effects of modifications at the histidine brace, just replacing the single helical peptide. And we have constructed three analogs, one with the regular histidine and another one with a methylation on the N-epsilon on the N-terminal histidine, which is a common modification found in fungal LPMOs. And further, acetylated version on the N-terminal amino group, which is intended to serve as a negative control of the importance of the amino terminal coordination on copper binding and catalytic properties. So we have investigated the oligomerization state of all the three analogs of DSB and by size exclusion chromatography. And here we can see that all of them show a single peak in the chromatogram corresponding to the desired heterodimeric form, even in the absence of copper ions. And then we have studied the coordination properties that I will not show in detail just for a matter of time, but we are currently studying the effects of modifications at the N-terminal histidine on the catalytic properties of DSB. And here I'm showing a schematic representation of the LPMO's catalytic mechanism, which could follow either oxygen activating pathway or a hydrogen peroxide activating pathway. So we first screen the ability of DSB and its analogs to activate molecular oxygen in the monoxygenase pathway, exploiting the formation of this intermediate, which is copper-2 hydroperoxide, which in the absence of external substrates releases hydrogen peroxide. So we have monitored the formation of hydrogen peroxide over time using a colorimetric assay. In particular, we used a peroxidase and a chromogenic substrate, in this case, the amplex thread. So here on the left side, we see the progress curves of peroxide release at pH 7 promoted by DSB analogs and free copper in solution. and on the right side, the dependency of initial rate of peroxide release as a function of pH. We can notice that the methylated analog shows the highest initial rate of peroxide release over all the pH screened, with the highest rate at neutral pH, pH 7. Then we have also explored the peroxide activating pathway by using the same model reaction that I showed you before for mini LPMO. And here we can see the progress curves that we acquired at pH 7, where, as expected, in green, the acetylated model shows the lowest activity, while the regular and the methyl analog seem to aim to have the same behavior. So seems that this modification does not affect this pathway, but just the oxygen activation pathway. However, we have gone deeper into this study and explored a range of hydrogen peroxide concentration. And here is a Michaelis-Menten plot. So we are reporting the initial rate of PMPG oxidation as a function of hydrogen peroxide concentration. And we can notice that even if the two analogs show similar K-cut values, they have very different KEM values, indicating a different affinity towards hydrogen peroxide. So we can conclude that the methylation decreases actually the affinity towards hydrogen peroxide, which also is consistent with the higher rate of peroxide release during the oxygen activation pathway. So just to summarize, we have designed and characterized two functional models of LPMOs, and our next plans are, of course, to complete the catalytic studies on DSB and its analogs, and to construct new models with incorporating some second coordination sphere interaction around the copper center, and integrating a carbohydrate binding domain to favor the interaction with natural polysaccharide substrates. So just a few seconds, I would like to thank all my colleagues from the Ambra Lab here at the University of Naples, all the people that collaborated to this project, and particularly Professor Angela Lombardi, who coordinated the research. And of course, thank you for your attention.
Thank you, Lisa. And there is time for a couple of questions.
Thank you so much. What a lovely talk. I'm here.
Thank you.
Do you see in the difference between the methylated and the non-methylated versions, do you see any difference in the rate of degradation of those proteins when you add peroxide?
Okay, I don't have a definitive question at the moment, but we are investigating this aspect because we do expect the methylation to protect the catalyst from oxidative deactivation, because it is one of the effects that it is demonstrated to have in natural LPMOs, which have higher turnover numbers when this modification is present.
Thank you very much for a really nice presentation. I will ask you only one simple question. Why did you use this kind of dye to check the hydrogen peroxide concentration? Because generally, I know that people use paratoluidine, but I see that I never see this particular dye, and I'm wondering if there is any, I don't know, any specific
no specific needs. Of course, we can use also other dyes, but yeah, we follow the literature procedure which involves this dye. We know the epsilon, so it's an established procedure. But in principle, one can use any other kind of peroxidase chromogenic substrate.
Okay, thank you very much.
Okay, so if there are no other questions, thanks Linda. And this is the end of this session. I would thank all the speakers that joined this session. Thank you, and maybe we see you 10 minutes.
This is the point for this, and this to go on and go back.
Oh, forward, backwards.
Yes.
Like that. Okay, great, thank you.
It's time to start. Please take your seats. So it's a nice pleasure for me to introduce Professor Anna Peacock. She grew up in the Netherlands and obtained her master chemistry degree from the University of York, where she was first introduced to the field of inorganic chemistry. She then pursued her PhD at the University of Edinburgh under the supervision of Professor Peter Sadler, followed by postdoctoral research with Professor Vincent Pecoraro at the University of Michigan. She then established an independent career at the University of Birmingham, where she's now professor of inorganic chemistry. Her research lies at the facing interface between chemistry and biology, focusing in particular on the design of application of peptide-based systems. And today she will present a lecture entitled Coils as Ligands for Inclusion in the Inorganic Chemistry's Toolbox for Advances in MRI Contrast Agent Designs. So, please.
Great, thank you so much for that kind introduction, and thank you for the organisers for inviting me to the beautiful city of Naples to tell you about our work in this absolutely beautiful venue. So I feel very honoured to be here. But I also appreciate the pressure with me being the speaker between you and the welcome reception. So I will do my best. What I want to tell you about today is the work that we're doing in my group to explore these kind of coils as ligands for use in synthetic and organic chemistry. And Linda just now gave a fantastic presentation where she showed a beautiful example of how they can be used to recreate catalytically relevant sites from biology with the copper hystrase. So we want to, in essence, normalize the use of these as ligands, but in the sort of toolbox that synthetic and organic chemists might frequent. And today I want to tell you about that work in the context of developing complexes that could have applications as potential MRI contrast agents. So in an organic chemistry, when we're developing new functional complexes, hopefully what we do is we go to the periodic table and we are excited by the breadth of choice that we have as inorganic chemists, and we're going to choose the element best suited for that given application. And so whether that's due to the coordination number properties of the metal, or the coordination geometry, or its reactivity, or its redox properties, we're going to select the element best suited. And then what we do is we use the toolbox that we have available to us to further modulate and enhance that particular property. And the toolbox comes to us in the form of ligands. And so our kind of toolbox in synthetic and organic chemistry is dominated primarily by what we might consider as traditional small molecule ligands. But if we look at nature, it has clearly evolved a really important class of ligands which come in the form of proteins. And these look very, very different from what we tend to use as synthetic and organic chemists. Now, these are much larger, but often they can modulate the chemistry of a metal ion in the way that we might struggle to reproduce a small molecule ligand. So there might be some opportunity here to learn from what nature has evolved in terms of ligand class. So I mentioned that what we're kind of interested in, or one of the applications we're exploring, is potential complexes for use as MRI contrast agents. And the reason we might want to do that is that we routinely use MRI in clinical diagnosis. So I had a look online, and apparently there's about 5 million MRI scans per year in Italy, about 40 million in the States. And in the majority of cases, those will be administered with a contrast agent to further aid in that clinical diagnosis. And most of the contrast agents are based on gadolinium. But there are some concerns about the use of gadolinium and contrast agents. It's a potentially rare, but it is a fatal condition linked to their use, which is NSF. There's increasing evidence of the retention of gadolinium complexes in the organs of patients, and they're now recognized as a contaminant of our water sources. And there's also that additional concern about the sources of gadolinium and geopolitical limitations on where you can actually access the gadolinium. So there is potentially an opportunity here to think about making new MRI contrast agents. Now, one solution would be to make more efficient gadolinium-based MRI contrast agents. If they're more efficient, you could administer less and in theory achieve the same level of performance, the same level of contrast. So if we take a look at some existing complexes that are used in MRI, and this is in no way an exhaustive list, we see that they have certain features in common. So they're based on gadolinium, and the metal has been chosen because it has 7 unpaired electrons. So that's really ideal. And in green, I've highlighted the ligands. Now, clearly the ligand does vary across this series. And like I said, it's not exhaustive, but the ligands have a lot in common. They really belong to the same kind of class of ligands if we look holistically at sort of ligand class design. And so if we're thinking about a new solution to gadolinium-based MRI contrast agents, maybe we need to think about a new solution to the ligand design. So on the left, we've got the traditional small molecule gadolinium complex. And on the right, we've got I suppose a traditional, but a natural metalloprotein complex. And these are clearly very, very different. But we and others, as we've just seen, are kind of keen in that space in between these two extremes. So using miniature artificial peptide scaffolds or artificial protein scaffolds as ligands for metal ions. And we saw, as I said, that lovely work by Linda and Angela Lombardi's group and so forth, looking at beautiful examples of biomimetic systems. So we set out to see if you could coordinate a lanthanide or gadolinium in particular to a coil, and then to evaluate the resulting complex in terms of its performance as an MRI contrast agent. So our first design is shown here. Well, actually, it's not our first design, but the first particularly successful design is shown here. And so what we opted for was a parallel 3-stranded coil, and we introduced a binding site in the interior of that design that we thought should be attractive for lanthanide coordination. So we've got 3 aspartic acid residues here, so a layer of aspartic acids, and then above that, a layer of asparagines. And what that should do is generate a -3 site, which is going to be complementary to the charge of our metal, but also present up to 9 oxygen donor atoms, which is typically attractive for lanthanide coordination. And then we made one further change, which is to introduce a tryptophan adjacent to that metal binding site. And that's for a couple of reasons. It basically just makes our life much, much easier. We can readily work out concentrations in solution, but it's also a really useful rapport to group of metal binding in close proximity. And in particular, if we were to bind terbium to this binding site or terbium in close proximity, tryptophan is known to be capable of sensitizing terbium emission if bound sufficiently close. So how does the peptide behave? Well, if we look at its folding in solution by CD, we actually see something that's very poorly folded. And that's because the binding site that we've introduced is extremely destabilizing. But when we titrate in our trivalent metal, what we see is that it induces folding and on coordination of the metal. So the metal here is behaving very much like a structural metal ion, so a bit like a zinc finger protein. So we can, of course, monitor binding indirectly by looking at sort of the degree of folding, but we can also monitor the metal directly in the case of something like terbium. So I mentioned that if we excite our tryptophan at 280 adjacent, if the terbium is bound sufficiently close, what we see is this nice sensitized terbium emission here. So in the absence of peptide, the terbium emission would be a little blip in the baseline there. And so this confirms it is binding in close proximity to that tryptophan. But in fact, the rate of decay of this signal is really informative, and it tells us about the hydration state of the bound terbium, because the OH vibrations are going to quench that emission. And so in this case, the data suggests that we have no waters bound to the bound terbium. Now, in some ways, that's consistent with our design. We've designed a metal binding site in the hydrophobic core of the kind of miniature protein scaffold, and the ligand scaffold can, in theory, provide all of the donor atoms, so there's no need to necessarily accommodate exogenous water. But as you'll see, we've been using gadolinium and terbium very much interchangeably. And the reason we do that is that in the majority of cases, gadolinium and terbium complexes generate the same sort of coordination chemistry. And so that's due to their very similar size. And here we've got crystal structures of gadolinium and terbium bound to this binding site within the interior of one of these 3-stranded coils. And largely the binding sites are as designed. And there's no evidence of any inner-sphere water. Now, granted, of course, the large size of the lanthanide is very likely to sort of swamp any electron density for the water. But in the case of the terbium, that's consistent with what we see in solution. And so you might wonder, why do I keep going on and on about the coordination chemistry and in particular the presence of bound water? But of course, when we're looking at MRI, what we're actually doing is imaging the relaxation time of protons. And most of those are found in water. So how the paramagnetic gadolinium interacts with bulk water is really, really key. And so the distance between water molecules, or the protons of water molecules, and the gadolinium is critical, whether it's directly bound water or second sphere, outer sphere. The rate of exchange between that water and the bulk is really, really key. And then finally, but in no way not significant, is the tumbling rate of that complex. So typically, large complexes that tumble more slowly tend to lead to higher performance in terms of MRI contrast generation at clinically relevant field strengths. So we decided to evaluate our gadolinium core coil shown on the right compared to deuterium, which is used in the clinic. And we wanted to evaluate it in terms of performance. And so the measure of performance I'm going to talk about is called relaxivity. And that's basically the efficiency of an MRI contrast agent. So the bigger your relaxivity, the more efficient it is, and you could administer less to achieve the same level of contrast. So deuterium, which is used in the clinic, has 1 inner sphere water bound to the gadolinium and is at 1.4 Tesla, so a clinically relevant field strength. It has these R1 and R2 as shown. Now, in contrast, our gadolinium core coil, we don't think has any inner sphere water. We think it's coordinatively saturated. So you can imagine we were quite pleasantly surprised when we actually saw a really competitive relaxivity. Okay, so that would suggest that you could administer less of such an agent to achieve the same level of contrast. So if it doesn't operate through an inner sphere mechanism, how does it work? Now, the complexes are, of course, larger, so it is going to tumble more slowly in solution, but there still has to be a mechanism by which we alter the relaxation time of that bulk water. And this is where we were really fortunate to work with Mauro Botta and his talented co-workers, Fabio and Marco, who I believe are here and who can answer any questions you might have on this work, because they did something called relaxometry, where you basically look at this performance measure, so look at relaxivity, as a function of field strength. And you can see how it changes as a function of field strength. And they fit these beautiful curves to some complicated equations, and they assure me that this is consistent instead with not inner sphere water, but rather 2 second sphere waters. And so what we think was happening is, in combination with, of course, the slower tumbling of our large molecule, but the hydrogen bonding of the second sphere water, like the core coil is basically encapsulating that second sphere water around that binding site, and then coupled with that slow tumbling. And that's responsible, we think, for this just quite significant uplift in relaxivity on going from the traditional ligands to these coil type ligands. So one problem with what I've presented to you is this coil-coil complex has a really poor stability. The log K is about 5. Okay, so this is not going to be relevant in it or useful in a clinical type setting. And one of the reasons for that problem is that we have these 3 separate strands that are destabilized, and they've got to come together. So unstructured peptides, to some extent, have to come together and form a really, really rigid pre-organized structure. So there's a huge barrier to assembly. So one thing we thought we could do is, instead of working with 3 separate strands, we can instead work with one long strand. So if we look at a helical bundle type design based on our original design, what we now have is a helix, and then in orange, the loop regions, and then a helix, and then a loop and a helix. So it's a bit more like a real protein, where you'd have your one long strand that folds into one structure. And so when we do that, and we look at how well folded it is, what we see is that even in the absence of a metal, we've got a really nicely folded assembly. Adding that metal leads to relatively small structural changes only, and so that this now leads to a higher affinity. So the log K has gone up to 7, so from 5 to 7. But that's still kind of a small change. That's not the step change that we need if we want to think about really utilizing these kind of complexes. So going from 3 separate strands to 1 strand is advantageous, but we need more if we want to pre-organize this binding site, if we want to really significantly increase the affinity for this kind of ligand. So what we reasoned is, if we take a top-down view, so this is looking at the individual 3 individual strands, but it's like a top-down view. And what we actually find is that at the helical interface, we rely on these salt bridges in part for the assembly. And so if we were to replace those salt bridges, in fact, with basically a covalent link. Okay, so if we were to replace them with these amide crosslinks here, maybe we not only have solved the 3-to-1 issue, but we've also pre-organized that metal binding site and rigidified it for metal binding. And so when we now compare the affinity, the terbium binding constants to these two different ligands, so the uncross-linked and the cross-linked ligand, we see we go from a log K of about 5 to a log K of 14. And that's just by introducing one single chemical cross-link across that binding site. And so that's starting to look like the step change that we might need to see. So if we introduce this change, what is the impact on performance? And so just to remind you, the relaxivity of our uncross-linked systems are shown on the left, but what you'll see is that when we introduce this relatively small change, it actually leads to quite a significant enhancement in performance and relaxivity. So what is the origin of that increase in relaxivity? And so in collaboration with Adrian Mulholland at the University of Bristol and his postdoc Marco, they did some MD simulations to try and understand the water access to this site. And so in the uncross-linked system, they can see that second sphere water that was proposed at about the right distance, and it's hydrogen bonded with that first coordination sphere, as you might expect. And we see the same water molecule in the cross-linked system, but this now has an additional hydrogen bond with that chemical cross-link. So with either through stabilizing it through this additional hydrogen bond, or merely just by blocking its access or its release from that binding site, we're proposing that what we're changing is the residency time of that second sphere water molecule. So using the coil ligand and the fact that we are thinking about the coordination sphere much beyond that first coordination sphere means that we can also start to control to some extent the behavior of second sphere water molecules, and that leads to a significant enhancement or change in performance, in this case in a positive way. So we've looked at the coil and we've demonstrated that you can generate lanthanide binding sites, and actually when you do that, this ligand does lead to quite a significant uplift in relaxivity, if that's the measure that we're interested in. And we can increase stability through chemical cross-linking and the like. But if we're thinking about ligand design, we want to use the ligands to control and to tune the coordination chemistry of the metal, because by controlling the coordination chemistry, we will tune the properties here in terms of MRI, but one could argue that applies to any kind of property you might be interested in, say catalysis. And so if we think again about the inspiration for this ligand, it's come from biology. So what are the tricks that nature uses? What are the tricks that proteins use to control performance of different metal sites? So one opportunity is to explore where you put that binding site within that coil. So here are the 4 potential locations for this otherwise identical binding site. So in yellow is the one I've just told you about. Okay, and so we can either translate, in essence, an identical binding site up or down that coil. So if we were to introduce it 1 nanometer below in this sort of red highlighted region, we find the chemistry is pretty much unchanged. There are small differences, but largely it behaves in the same way. But if the binding site is introduced towards a terminus, so towards the C terminus at the bottom of the coil, or towards the N terminus at the top of the coil, we start to access hydrated metal sites. And that's not really surprising, because what you can imagine is that this coordination site, not all of the donor atoms are fully engaged in coordinating to the metal at all times. Water could come in and temporarily populate those. But of course, at the end of the coils, you could imagine expansion, fraying, and water access is improved compared to the more buried central positions here. And in fact, in the top binding site, and this is merely a model, it's not we haven't got structural information for this, unfortunately, but we propose that those asparagines aren't involved in coordinating at all, and that they permanently vacate those positions. And in fact, if we remove the asparagines, you retain the ability to bind. So translating the binding site is another way by which you can control the chemistry and ultimately its properties. But you could also think of changes beyond the first coordination sphere. Of course, changing the first coordination sphere ligand set is going to have an impact on the coordination chemistry, but we can also take advantages of those residues that are in close proximity. We've just seen how the cross-link can change the chemistry of the site, and that was not even necessarily a second sphere residue. So what we did is we took the top binding site. So for simplicity, or hopefully for simplicity, I've just shown you one part of one helix. Okay, so this is the top binding site, and this was the highly hydrated site. So we think there are 3 waters bound to the terbium. So it's got an aspartic acid layer, an asparagine layer, and above that an isoleucine, so a non-coordinating residue. And so we were interested to see what would happen if we systematically alter the identity of that residue, increasing its steric bulk. So going from isoleucine to phenylalanine to tyrosine, and ultimately to tryptophan. And as we increase the steric bulk of that second sphere residue, we lead to a systematic decrease in the hydration state of that bound terbium. If we go the other way, if we try and remove steric bulk, what we find is that we don't increase the hydration state further. So if we present both the aspartic acid and the asparagine, then the maximum hydration state appears to be 3. But what that means now is that we've got, if we take advantage of the full versatility of the coil scaffold, such as first coordination sphere residues, absolutely, but also binding site location and second coordination sphere residues, we start to be able to predictably tune in what hydration state we want. So we can talk about coordinatively saturated sites, which are the examples I've been discussing, but we can go all the way to extremely hydrated sites. And if we can change the coordination chemistry, we can change the properties. In the context of what I'm telling you about, we're looking at the gadolinium complexes over there, and looking at relaxivity as the performance indicator, but I'd argue that this applies to other properties as well. And so when we increase the hydration state, not surprisingly, we increase the performance, we increase the relaxivity. So you can see we can start to tap into a really large range of different performance measures. Okay, so one of the things that the coil I think sort of shouts out for is the opportunity to embed more than one functional site into the design. And so initially how we decided to do that is we wanted to embed two different metals, but we wanted to embed different metals, and importantly, we wanted to do so with control and selectivity. So we didn't want to have the same metal, and we didn't want to have a lack of control about where they went. So initially what we did is we took advantage of the fact that our lanthanides are very hard, and so we took one of our hard lanthanide binding sites, and on the other side of the coil, so kind of far removed from that first binding site, we introduced a very different binding site, a soft binding site for a soft metal. And so not surprisingly, the terbium goes to the terbium binding site, and the mercury goes to the thiol binding sites, and we see those spectroscopic signatures associated with those individual sites. And so they behave quite independently of one another. And that's fine. So we have control and selectivity, but I think it's fair to say this isn't the biggest challenge, having a hard metal go to a hard site and a soft metal go to a soft site. What we really want to do is to be able to bind to two very similar metals, but again with that control and selectivity to two separate sites. And in particular, we want to bind to two different lanthanides. So we've already seen that different lanthanides have different properties. Okay, we've been talking about gadolinium in the context of MRI, but we've been using terbium because of its visible emission. So there are lots of appealing properties across the lanthanides, but the problem is that their coordination chemistry tends to be really, really similar. So if we want to achieve a goal like this, we have to find a way by which we can discriminate between lanthanides for different sites. And so we took, and I don't want to call it a library, it's more of a bookend. We took a couple of binding sites, 5 in fact, and we were keen to see what would happen, and could we discriminate between the lanthanides based on their size? Because though their coordination chemistries tend to be very similar, the key difference really is the size as you go across the series. So what we did is we loaded up our binding site with terbium, and we looked at that tryptophan sensitized emission for the bound terbium, and then we added in a competing lanthanide and looked at displacement of that original bound terbium. So when we add in gadolinium, which is the one we use interchangeably, we see pretty much 50% displacement across the series, and that's consistent with gadolinium and terbium having really similar coordination chemistry. But if we look at the remaining lanthanides, what we see is kind of two notable things. First of all, binding sites at the top of the coil are really fairly promiscuous, and they'll pretty much bind to any lanthanide they're given with no notable preferences. Whereas the more buried binding sites have a preference for these kind of medium to larger lanthanides and discriminate against smaller lanthanides like ytterbium. So we should be able to combine a selective site with a promiscuous site and generate a coil that can bind to two different lanthanides with this control and this selectivity. And so if we look at the binding of lanthanides to this particular peptide, we can confirm it forms a well-folded coil, and we can confirm that it does in fact bind to two lanthanides. But of course, that doesn't tell us anything about control or selectivity. In order to interrogate that, we prepared the analogous peptides that lack, or in fact, they contain just a single tryptophan reporter group adjacent to one site. So this tryptophan is going to report on binding of terbium to this site, but is unable to report on binding to the second site. And here we have the reverse. And so if we titrate terbium into this peptide on the left, we see an increase in emission of terbium emission from 0 to 1 equivalent before it basically plateaus. And in contrast, when we do the experiment with our control peptide on the right, we see the large increase really from 1 to 2 equivalences of terbium before plateauing. So this strongly suggests that terbium is primarily binding to our size selective site, and only when that's pretty much populated does it really populate our second site, which is the one that's promiscuous and can bind to ytterbium. And so not only can we think about control and selectivity, but we can also change the position of our size selective site. So we can translate that site along the coil so we can get this control and the selectivity, but also now start to think about distance-dependent communication between those two sites. Okay, and I just want to show this. It's not one of the peptides I've shown you, but this is one of our dual lanthanide binding coiled coils because we don't get very many crystal structures. So when we do, I have to share them. And in fact, Valentina Borghesani, who's in the audience, this is some of the work that she did when she was in my group, and she's a very talented chemist at the moment at the University of Parma. So one of the solutions to the MRI contrast agent challenge is to think about making more efficient gadolinium complexes. Another solution is to replace the gadolinium entirely. And there's some beautiful work looking at manganese and iron to some extent as well. But we started to think about actually what other metals could you use? Because what we ideally want to use are biologically essential metals. Because if you can use a paramagnetic biologically essential metal, that's going to alleviate many of those concerns that we've just mentioned. And so for a completely unrelated reason, we started to think about copper. And we're interested in copper because copper is widely used by biology for loads of different functions, and its coordination chemistry, not surprisingly, is very diverse in biology. And this is not in any way exhaustive, but you have examples of copper sites bound exclusively through sulfurs, exclusively through nitrogens. We saw some nice copper nitrogen sites just now. Lots of mixed donor sites, so copper's bound to sulfurs, nitrogens, and oxygens. But as far as I can tell, not a single example of a copper bound to a protein scaffold exclusively through oxygen donor atoms. Now, there'll be a perfectly good reason for that, but if you think about synthetic inorganic chemistry of copper, there are lots of nice examples of copper complexes bound exclusively to oxygen. So we really were keen on seeing how we could generate such a site and ultimately what kind of properties such a site might have. And we reasoned that the ligand I've been telling you about would be the perfect scaffold with which to generate it because we haven't got any histidines or cysteines or methionines. What we have is an oxygen-rich binding site that isn't biased towards a particular coordination geometry or number. And so in theory, the metal could pick out the donor atoms it wanted. And so we interrogated how this system, how copper binds to this peptide. We looked at folding by CD, we looked at fluorescence quenching, we did some XAFs, and everything points towards the fact that copper is binding and it's binding exclusively to oxygen donor atoms. But we were curious as to what kind of properties such a copper might have, what kind of properties such a site might have. First, we thought maybe catalysis, and if anyone's interested in exploring that with us, please do reach out. But given the work we've been doing, and I'm sure you can see where I'm going with this, we were curious as to whether there was any potential from an MRI perspective, because copper in the 2-oxidation state is, of course, also paramagnetic. And so we decided to evaluate our copper coiled coil shown in blue now, it's the same ligand, compared to our gadolinium coiled coil. And just for context, I'm giving you the values for deuterium in terms of performance, okay? So these were the relaxivity values I showed you earlier for deuterium or for our gadolinium coiled coil at 1.4 Tesla. Now, these are based on gadolinium because gadolinium has 7 unpaired electrons. That's why gadolinium is used in MRI contrast agents. In contrast, we are now asking our system, our complex, with a copper-2 that has 1 unpaired electron. And presumably for this reason, copper-2 has been basically almost entirely disregarded for use in MRI contrast agent design, because 1 electron compared to 7, I think we can all appreciate there's a significant difference there. And in fact, if you look in the literature, there are only a handful of examples where people measure the relaxivity of copper complexes, and they're really very, very low. So, setting the scene, but basically we were pleasantly surprised to find that actually our copper coiled coil has really, really very competitive relaxivity. In fact, the R1 is comparable to that of our gadolinium coiled coil, and the R2 is in fact superior. So though this may not be the complex that is injected as an MRI contrast agent, what it certainly does is it challenges the idea that copper isn't suitable for use, that it isn't capable of high relaxivity. It absolutely can. Copper-2 complexes can achieve really high relaxivity. So what is the origin of the performance of this coiled coil? Is it exactly the same as what we saw for our gadolinium coiled coil? Does the ligand do exactly the same? And the answer, of course, is no, it's not, because the copper coordination chemistry, not surprisingly, is going to be really, really different to that of gadolinium. So again, Mauro, Fabio, and Marco come to the rescue, and they perform relaxometry measurements on this copper coiled coil. And if you recall the data previously from gadolinium, you'll recall that this looks really very, very different. And in fact, this is apparently better described by a model where we now have 2 inner sphere waters. So not surprisingly, the different coordination preferences have still generated a different complex, even though we presented the same ligand sphere around it, and how it interacts with water is really different. And so the origin of the relaxivity for this complex is a combination of those two inner sphere waters, presumably coupled with, of course, the slower tumbling in solution. So hopefully what I've done is convince you that the coil illustrated here warrants inclusion in this toolbox of ligands that we have available to us across synthetic and organic chemistry, not necessarily limited to biologically relevant sites or biologically relevant applications, but actually across inorganic chemistry much more widely. It can be used to, the coordination chemistry of the metal, which is what we care about if we're interested in performance, can be tuned by things like binding site location. It is sensitive, not surprisingly, to first coordination sphere residues, but also to those residues positioned in the second sphere. You really got a very versatile ligand that you can use to tune the chemistry of the bound metal. We can use it to distinguish between what are otherwise really similar metal ions. So the lanthanides are very similar and they differ really in their size. We can bind multiple metals in these kind of coil scaffolds. And in terms of the application I've told you about, the coordination of a coil to tried and tested gadolinium leads to a significant increase in performance, but it even leads to a significant increase in performance for copper, which is the opposite of tried and tested, maybe neglected. And so that just challenged the idea of maybe copper-2 is suitable for use, maybe not in a coil, but is certainly capable of high relaxivity and warrants consideration for future MRI contrast agent design. So I just want to thank the key people involved. Kate Hadley has done most of the work I'll probably be presented. Sarah and Kate Kogan are new to the group. Very grateful for my collaborators who I've mentioned our funding. And of course, delighted to thank you again for your very, very kind attention. Thank you so much.
And there is time for questions. Okay, we have a question.
Thank you for the lovely talk. This behavior of copper is very surprising in light of the small magnetic moment. So I didn't really get how general this is. So is it strictly dependent on your specific ligands, or what are the secrets there?
Okay, so that's a fantastic question, which, as you can appreciate, we're trying to understand. So the small molecule complexes that I've seen reported have very low relaxivity. And I have to admit that all the variations we've explored with copper based on a coil have a lower performance than what I've presented here. Some of them are still relatively quite good, but not as high as here. So everything we've done so far has managed to make it worse, not better. So we are, as a result, starting to maybe understand, but we are really probably far away from fully understanding why. So if you have any ideas, please share.
Barbara? No?
Thank you, very nice talk. Maybe it's not your main goal, but have you ever tried to see if this current environment allows the lanthanides FF transition to be optically active by circular reason?
So we haven't explored that as much as we'd like to, but you're absolutely right. That's one of the things on the list of things that we'd like to explore. Yeah, it's a great idea.
Thank you.
Well, thank you very much. Lovely talk. I know nothing about coils. Very hard to pronounce, by the way. So have you ever, so all the examples of structures that you've shown today have, with lanthanides at least, have 3 coils. Have you ever tried with less than 3 coils? For example, 2 coils. Are they able to efficiently avoid solvent coordination, or do you get solvent around?
So we haven't, because we felt that the 3 coils were important, as you've suggested, to make a stable site. It gives us that 3- charge and it protects us from the solvent, because we did want to have some control. There is some work in the literature a while ago. Hodges looked at 2-stranded coils where they had negative charges that prevented the coils assembling and adding in divalent metals, but also they did look at lanthanides, help them assemble. So you can generate binding sites at those at that helical interface, but one would imagine they'd be very hydrated, and maybe it might not allow you to have that water exchange rate that you want, but you can generate them. They're just generally less specific if you put them on at the helical interface.
Thank you.
Some other question? Okay.
Oh, thank you. You mentioned it, but I'm curious, have you tried manganese? Because manganese too can have a relaxivity very even surprisingly.
Yes, we have, and it's fabulous. So yes. It's very warm up here. It's probably very warm for all of you as well.
Some other question?
Thank you for the wonderful and very interesting talk. I have a practical question. You made some linkage between coils, and I'm wondering, when you make a coil, you have several aspartic or glutamic acid and several lysine. So you make just one or more than one cross-linking between the whole bundle, and if you make more than one, how can you distinguish between maybe the exact one and the shifted between coils?
So if we were to do non-specific cross-linking, we've observed what you described. But in this case, we introduce a modified glutamic acid in the synthesis, so with a different protecting group that we could selectively deprotect and activate. So we only have one glutamic acid that cross-links. And we verified that it only cross-links with one lysine, not the other lysine, because in theory, distance-wise, they kind of should, with mass spectrometry sequencing and proteolysis and so forth. So because we introduce a non-natural amino acid specifically for that glutamic acid. But you're right, when we do non-specific cross-linking, it is non-specific and we get a mixture.
Okay, thank you so much.
Thank you. Wonderful talk. Connected to the question from this side about the number of coils, since formally copper has another oxidation state than gadolinium. So for gadolinium 3, you need 3 coils to compensate for simply electrostatics reasons. Have you tried for copper 2 coils and a bimetallic approach for manganese, connected to the other question, that works wonderfully with copper, for example, like as you did with gadolinium and the other lanthanide?
Yeah. So in answer to your first question, no, we haven't looked at 2-stranded coils for copper. We've only been looking at these 3-stranded systems to date, but we are very much interested in going beyond 3-stranded, but probably more like 4-stranded. I think 2-stranded, you haven't got the same control of the metal coordination chemistry. It's different when you're trying to sandwich a heme between 2 helices. But if you've got a metal binding site, it's very exposed and it tends to not be very stable or protected. So we tend to avoid that. The bimetallic, yes, absolutely. We're very interested in basically all the combinations, and that would be a lovely one to consider. So, yeah, great suggestion.
Thank you.
Thank you for the talk. I was wondering, apart from the relaxation, which I found really interesting that it goes up instead of down, but do you expect also some influence on the localization of gadolinium inside the body? Is that something that you were looking for?
So, in all honesty, we haven't done anything in vivo with these systems. And so one of the big things we need to do now is to see how they behave. There are examples, or there is an example of a coil that's been explored for a completely different application in clinical trials. So in theory, I think you can mask the outside to be tolerated, but we haven't explored it. I guess the theory is that because it's peptide-based and sort of protein-inspired, you can easily add in things like nuclear localization sequences, either on the exterior or on the termini. And so bringing in those opportunities that peptides afford for targeting should be compatible with this kind of design. But no, we haven't actually done that yet. But we would love to.
Thank you.
So thanks to Anna for a wonderful presentation and the wonderful results too. And I would like to remember that at 7:30 we have the welcome party waiting for us at the Royal Continental, and so we have 20 minutes.